Good morning, everyone. Thank you all very much for joining us. We're going to go ahead and get started. My name is Nick Murdoch. I'm a research associate at the Forum for Collaborative Research, and I think I've been in contact with most of you as you've gotten registered and prepared to join us today. Thank you for coming today to our HIV treatment bNAbs Clinical Research Considerations workshop. I'll be starting with welcome, housekeeping, and a forum overview, and then I'll turn it over to our director, Veronica Miller, for some more introductory remarks. To begin with some housekeeping for our in-person attendees, please note that breakfast, lunch, and drinks will be available in the lobby. You're welcome to bring any food or drinks into this room, but we'll be serving everything in the lobby. Bathrooms in this building are located just out these back doors in the right of the room, past the coat rack, and then on your right. You're welcome to use the coat rack. If you have any luggage that you'd like to store, you can place it behind this table that we have in the back. Forum staff will be at this table throughout the meeting. We do have some participants joining us on Zoom because of pre-existing time conflicts. So when you speak during this meeting, please make sure that you speak into one of the microphones. If you're on the stage, no need to touch any of the microphones. They're all already set up. At the podium, obviously, we have this one. And then if you're in the audience during a Q&A period or you have a comment on a presentation, please stand up and come to one of these question and answer microphones that we have placed in the center of the room. All microphones are on at all times, the way that this room is configured. You have been warned. So if you need to have a private conversation, please make sure you step into the hallway. Otherwise, you do risk potentially a Zoom participant hearing what you're saying on the phone. And you might notice that this meeting is being recorded. That is just for note-taking purposes. The recording of this meeting will be used only internally with the forum and will not be distributed online. And with that, I'd like to begin with an overview of the forum itself. So the HIV Forum is a platform for ongoing multi-stakeholder dialogue so that we can advance and accelerate treatment and prevention development for HIV. We do so by providing a neutral, independent, and safe space for discussions and deliberations across stakeholder groups. And we focus on developing consensus by doing multiple working group meetings throughout the year. Many of you have joined us on Zoom previously, and we do larger meetings such as this one in person. And we function, most importantly, based off of active and engaged participation from all of our stakeholders, which is why we're very happy to have you all joining us in the room or on the Zoom here today. The forum's concept is that by getting everyone, regulators, patients, industry, and academic experts at one table, we can increase efficiency by creating enhanced clarity, more innovation, and greater collaboration, and decrease uncertainty by reducing redundancy, reducing development time, and reducing risk. Our operating principles is that the forum is an independent and neutral venue for ongoing dialogue. Each stakeholder group has an equal voice here. We are here to facilitate the best science-based decisions in real time, and we do so by breaking down inefficiencies and redundancy to increase clarity, collaboration, and innovation to benefit the whole field, most especially patients. The forum is a safe space. This is a closed meeting with no marketing staff and no press. So please bring your expertise and leave your hat at the door. The rules of the room and Zoom are that what is said in this room stays in this room. It is not meant for attribution. We ask that you speak from your expertise and that you let the science speak. And we ask that everyone involved, whether here in person or I'd like to speak especially to our participants on Zoom, that everyone is active in the discussion and that you feel you can use your voice in this space. I'd like to thank the HIV Forum sponsors, Gilead, Merck, and ViiV, and I'd also like to note that our PrEP project has additional support from the Bill and Melinda Gates Foundation. I would also like to give a special thanks to our forum team. Our director, Veronica Miller, is here and will be speaking shortly. The bolded names here are our HIV Forum staff, myself, Logan Donaldson, who is here in the side of the room and will be available throughout the day. Our colleague Robin Schaefer, who has just started parental leave. And I'd also like to give a special thanks to our two newest staff members here, Christopher Berry, who is our events manager in the back of the room, also available if you have any questions or needs throughout the day, and Denise Butler, who our in-person attendees saw at the front desk today. Thank you to all the forum staff for pulling this off. And now I'd like to turn it over to our director, Veronica Miller.
Thanks, Nick, and welcome everyone. I'll just provide a few additional remarks on what we want to achieve here today. We have a good representation from all the stakeholder groups. So again, welcome everyone for coming here. So the purpose of this meeting today is to start this discussion on what is the path for approval and then also implementation into clinical practice if approval is granted of broadly neutralizing antibodies as an additional therapeutic to treat people with HIV infection. There's, of course, other bNAb research going on in the prevention area, mother-to-child transmission, cure research, and Joe Aaron will talk us through these other, well, not talk us through the scenarios, but just kind of put what we're doing here today into perspective. So at the forum, what we like to do, I often refer to it as a deconstruct, reconstruct exercise. So today we will start off with sort of, this is the overall overview of what bNAbs are and where they might function, but then we'll zero in on this is the therapeutic area. And then from there, what we really want to discuss today is what is the role of diagnostics in antibody neutralizing activity in terms of designing a clinical trial, recruiting patients and entering patients into a clinical trial. And then, of course, also we will be very interested in the outcome in terms of what happens to the viruses of patients that are not completely suppressed. But that's really, so it's a very specific part of the whole puzzle. And this is just the first in a series of meetings that we will sponsor on this topic. So yes, we want to always keep the big picture in mind, but then really focus in on the very specifics as outlined in today's agenda. And so this is really what Nick has outlined in these slides here today. I don't want to take too much more time, but you know, it's okay to expand a little bit, but then we'll always make an effort to bring it back down into the focus for today. And we do also, for this meeting today, have a US focus. We have you know, great participation from our colleagues from the US FDA. We also work a lot with our European collaborators and our European collaborators at the European Medicines Agency in different countries. But at this particular time for today, we will focus on the US path. And with that, I guess we call up the first talk with Joe Aaron, who will give us the overall context. Thanks.
Great, thank you, Veronica. Don't know. I guess you guys cannot see. Oh, you can sort of see a pointer. All right. So thank you very much. I'm really going to give a very broad and short overview of where I think bNAbs may fit into our treatment landscape, and then make a nod to the use and potentially in cure strategies. So I think the main area for bNAbs is the issue of very long-acting therapy. So the setting is there are some molecules that are very long-acting, small molecules, but they lack partners. And a substantial portion of people with HIV really on treatment desire long-acting therapy. And every time I talk to another patient, there seems to be another reason why: stigma, anxiety. They can't remember whether they took the pill or not, and that causes problems for them. They may actually be threatened or uncomfortable disclosing that they have HIV. We haven't really addressed this with long-acting therapy, but people who can't access therapy, can't navigate oral therapy, or are not reached by our current system are people who may be benefit from very long-acting therapy. And I think 95 is very optimistic for the US, but we need to reach those last 5 or 10%. And then the populations are people who are suppressed on therapy, on oral therapy, or we haven't really explored yet people who are naive or viremic. So there's several different ways that this might play out, and probably people in the audience could think of other ways in addition. Obviously, bNAbs combined with small molecules, and I'll give some examples of that. That has some positives and potential negatives, but they certainly need to have similar half-lives, and I'll show you an example of bNAbs with lenacapavir, but there are obviously other projects going forward. Pure bNAb combinations. Question is, well, how many do you need? And they likely would need different targets. So we'll talk about that. There are companies developing tri or quad specific antibodies. And then you could combine bNAbs with other long-acting antibodies. And there's an example of that that's under development. This is the lenacapavir with 2 bNAbs, teropivimab and zinlarivimab, that Gilead sponsored. This was a trial of 2 bNAbs plus lenacapavir. It reported susceptibility to both bNAbs, and it really was an initial proof of principle trial in people who've been suppressed, and most had been suppressed for a long period of time. It looked at 2 different doses of the zinlarivimab, but it really was kind of a proof of principle trial. There were about 120 people that were screened. 12% or 15 couldn't be susceptibility. The susceptibility test wasn't completed, and about half of the remaining, almost exactly half of the remaining people turned out to be eligible. But what we did show is that it worked. There were 20, if you look at the 2 different doses of zinlarivimab, 2 people did not achieve suppression at 26 weeks. 1 left the study while suppressed, and another did rebound at a low level, and resistance testing is still ongoing with that particular person. But the concept was, I guess, proven over the short term. There is a pilot study where individuals who screened for the study that were sensitive only to a single antibody were actually treated with the 2 antibodies and lenacapavir. Those data are pending and hopefully will get shared at a conference that's coming up. So those data may help inform whether susceptibility to 2 antibodies is required in this setting. So there are other examples. ACTG did 5357, which was VRC07 523LS with cabotegravir, that I know has been submitted to CROI. I mentioned the teropivimab, zinlarivimab, lenacapavir. Now the phase 2b study is recruiting. In fact, it's at least reached its full screening capacity. This is a true phase 2b study with every 6-monthly therapy compared to continued oral therapy. N6 is being developed by ViiV plus cabotegravir. This is also in a phase 2b study that's recruiting currently at multiple sites. I feel bad. I misquoted Boris, but turns out I was prescient. Boris Yulg, who's in the back of the room, studied 3 antibodies in viremic individuals, and that study has been published. I searched inadequately yesterday when I was making these slides. And then I understand from him that there's also a study ongoing of these 3 antibodies, and people are suppressed. And then there's also a study of a 2nd generation ibalizumab, so another antibody, but not a bNAb, combined with VRC07 523LS and suppressed people, that's ongoing. And there may be other examples, but these are the kind of things that we're going to see, I think, over the next short term. The hurdles, which is what we're going to talk about today, There's susceptibility testing versus very broadly acting combinations. There's administration. Right now, the amount of antibody that needs to be infused is probably not going to work well for subQ dosing, but I know some companies are exploring that. There's need for probably multiple antibodies or complex structures or partnering with small molecules. And I think partnering with small molecules puts the small molecule at risk, which is I think has different consequences depending on the small molecule. We're going to talk about ADA. It's kind of interesting because ADA might emerge later. When we give people oral therapy, we think, well, once you get suppressed, and if you've been suppressed for a while, then your likelihood of failure is very low. On the other hand, with ADA, that could be a late phenomena. And this is really not a comment on pharma. Their preference is obviously for the sure thing, right? People are suppressed. Can we keep them suppressed as opposed to the hard thing? And I understand that they have a job to do. But almost all initial studies are going to be done in people with HIV who are suppressed, in care, and adherent. And that may not be the group that needs these antibodies. This is just reminding people that we're nowhere near 95 in the US. The CDC thinks maybe 62% of people have sustained suppression. And there was the Lancet HIV article that is on the right. If you look in Southern Africa, there's some shining examples of sustained suppression. But if you look across the globe, the estimate is somewhere around 65% of people are in care and have sustained suppression. The other thing that I haven't heard talked about, but I saw Tae-Wook Chung here somewhere, this idea of treatment for those who have untreatable MDR TB and MDR HIV, sorry, and we should talk about that a little bit at some point. In other words, people with resistance or multi-agent intolerance and limited treatment options. And perhaps we should also urgently study these antibodies in those people. We do really crazy things to treat small populations, like, for example, pig hearts that cost hundreds of thousands, if not millions of dollars. And these are people, a very small fraction of people that need therapy. And maybe we should think about compassionate access to bNAbs for those who are untreatable. And then I just want to mention, because we have talked about this, bNAbs are also being explored as a component of functional cure or reservoir reduction strategies. And I just made the slide yesterday with a bunch of different references that people can look at. But obviously there's data in small models of SHIV or SIV where antibodies seem to lead to prolonged suppression. And maybe also in people, and that may be due to a vaccinal effect. They may induce some special T cell related immunity, and certainly that's scientifically interesting and perhaps practical at some level. And of course, maybe they could be used to clear infected cells or target other entities like CAR Ts or these bispecific bites or darts to infected cells as part of functional cure. But I think that the main message is that bNAbs give us an opportunity for very long-acting therapy and can be used in combination. And when therapy is spaced by 6 months or more, perhaps the mode of infusion or the mode of administration is not as big a hurdle. I did have a slide. I forget who sent it to me. Somebody from oh no, it's from someone from the VTN. Now I'm not remembering who, where he showed me these infusion centers in Johannesburg where you can like go after a tough night in the town and go and get an infusion of saline or vitamins or just on the street. So infusions are possible. Anyway, that's my summary, and I don't know if we're taking questions or moving on. If people have questions, I'm happy to try to answer them.
We can have one or two questions. And thinking about that infusion center, I'm just thinking about, the increased utility of bNAbs in healthcare generally, and all of the different autoimmune diseases, and in, beginning of the COVID pandemic, a lot of bNAbs were used and might be used again for other viral outbreaks initially. So what do you think we need to watch out for just in general? Will people's arteries get clogged up with with antibodies at some point?
And actually, one of the things that I left out that I should have included is the remarkable safety so far of these virally targeted, broadly neutralizing antibodies, whether you're talking about COVID antibodies or whether you're talking about bNAbs. And if you think of the AMP studies, or now they're just remarkably safe. Certainly, there are occasional descriptions of adverse infusion reactions, but they're really uncommon, which is very different than some of the autoimmune antibodies where ADA and infusion problems, et cetera, are much bigger. I think the issue of they go in easily, they're not toxic. I think people could sustain for a long time. I mean, obviously other modes of administration like sub-Q administration would be better. And other people here that are, more experienced than me could talk about what are the challenges of kind of sub-Q administration. I know that the volumes, at least so far, I mean, we're talking about, 20 or 30 milligrams per kilogram. That's a lot of antibody. I think if as potency goes up, then infusion volumes will go down. That makes a difference. But
great. Maybe they could give them on an airplane. I mean, if people are going to be stuck for several hours.
all right, well,
that's maybe a little bit too much, but this is a great overview. Any other comments or questions for Joe specifically? Oh, we have a question on Zoom. Okay.
Wait. This is our chat. I'm sorry. I realized I was. Oh, you can unmute on Zoom. Sure, if you have a question. Yeah, that would be
wonderful.
Probably the better idea.
Thank you. And sorry I couldn't join you today. My name is Sharon Ann Lynch. I'm a co-director of the Center for Global Health Policy and Politics at Georgetown. And I have a question that I think at the tail of your presentation and before we hear from others I hope would be helpful, which is, given the high complexity and associated high cost of antibodies, what have we learned from private sector industry about their goals towards making the products affordable and suitable? We know from some subject matter experts that mAbs, so the monoclonal versions, could be made for as low as $50, as low as $25 a gram, as opposed to where they're at now, which is the sky. This would take some optimization efforts. And as of now, the private sector hasn't had, I guess, a perceived commercial incentive to explore or put in place some of those efforts to bend the cost curves, whether in terms of the product efficiency, lower dose, higher half-life, for example, more potency, requiring fewer administrations, et cetera, as well as some strategies on the manufacturing side. So there's some research happening now in terms of alternatives to the Chinese hamster ovary cells, other ways of manufacturing that might reduce costs. But what is happening now is could be the norm for the next 20 years. So the question is, given that low and middle income countries represent just 10% of the mAbs available, high income countries represent 80%, we need to tinker now to make sure that we optimize on the product and the manufacturing process as much as possible to create a new standard, a new norm that we hope is at a lower price. And of course, innovation is iterative and improvements can happen along the way. But that missing piece is so critical. So I hope the next panel, as well as yourself, can speak to that a bit. Over. Thank you.
Yeah, I'm sure that there are other people in the audience that would be better at speaking about that. My one small comment would be that we still don't really even know whether this strategy is going to work. I don't think we want to inhibit the proof of principle in smaller scale studies that show that it can work while waiting for more affordable and scalable treatments. But I agree, your points are excellent and should be part of the discussion at some point for sure,
Right.
maybe later today.
So thank you very much. And we, Sharon Ann, very good to have you here joining us by Zoom. And we invite all of our advocates and everyone participating to advocate for global access once a therapeutic is available. But for today's discussion, we will not be talking about that because we're really focusing on the question about the diagnostic requirements for doing these studies. So it's a small piece of the puzzle, but it will tie in. Obviously, if we can't figure this first part out, we don't have a product to optimize. So thank you for that comment. Let's move on then to the next part, which is a pre-recorded talk by Dan Koritsky, who is here, except that unfortunately the schedulers above were not kind in terms of another meeting he had to attend to at this very same time. So he's kindly pre-recorded his talk, and so we'll hear it, and then we'll hear from Jackie Reeves from Monogram, LabCorp Monogram Biosciences, about some of the lessons from the CCR5s. So there we go, Dan.
Good morning. I'm sorry to be giving my talk remotely. I'm actually here in Washington, but have a faculty meeting to select next year's fellows that I have to be present for, and I'll be joining you in person later in the day. Now I'm going to talk to you about measuring antiviral activity in antiretroviral drugs. The issue is really, what did we learn from our use of genotypic and phenotypic testing for traditional small molecule antiretrovirals, and how might that inform our discussion about the role of susceptibility testing in the use of broadly neutralizing antibodies for the treatment of HIV. These are my disclosures. So let's start with a discussion of genotypic testing for drug resistance. Genotyping, of course, determines the presence or absence of specific changes in HIV genes encoding the drug targets, such as capsid, protease, reverse transcriptase, integrase, or envelope. But because HIV is so variable, we can't simply take the presence of a nucleotide or amino acid substitution compared to some consensus sequence as evidence of resistance. The interpretation of a genotype presupposes knowledge of which are the critical mutations that actually confer resistance. And so we're really inferring drug resistance by the presence of these known mutations. Now, there are a variety of methods and platforms that can include Sanger sequencing or several of the next-gen sequencing approaches that may be used to generate this sequence information. But ultimately, these all require some kind of interpretive algorithm. Phenotyping, of course, measures directly the amount of drug necessary to inhibit viral replication in a tissue culture system. And this can be used to determine what the IC50 or the concentration needed to inhibit replication by 50%, or the IC90 by use typically of recombinant or pseudotype viruses. And these assays, especially when they are automated and done in highly replicatable manner, can be quite precise, measuring changes of just 1.5 to 4-fold. But the challenge here is interpreting the meaning of a shift in susceptibility. Clinically relevant breakpoints have not been determined for all of the antiretrovirals, and it's critical to remember that these assays merely measure drug susceptibility. the definition of drug resistance really requires clinical correlation. So there are many challenges in defining antiretroviral drug resistance. First of all, there are numerous reasons for the failure of ART. Not all of them have to do with resistance. There can be poor adherence, inadequate regimen potency. There may be pharmacologic factors that lead to inadequate exposure. And of course, there can be either transmitted or emergent drug resistance. There are then several steps that are needed to define antiretroviral drug resistance. Typically, this begins with the emergence of particular mutations in association with virologic failure in people being treated with the drug in early phase clinical trials, then demonstrating that the mutations observed actually are responsible for reducing drug susceptibility by introducing them through site-directed mutagenesis into recombinant viruses and reproducing the phenotype. It then becomes important to demonstrate that the presence of that mutation actually impairs treatment response and to determine the fold change in susceptibility that's associated with reduced clinical efficacy. Now, this slide takes us back nearly 30 years to the very first study that demonstrated the clinical significance of a shift in susceptibility to zidovudine. This was a study done by Rich D'Aquila, the late Vicki Johnson, Tony Japour, Clyde Crumpacker, Doug Richmond, and myself, using samples collected from a study that compared continued zidovudine to a switch to didanosine in zidovudine experienced people. This was ACDG 116B 117. And we had developed, with the help of Doug Mayer, a consensus phenotype assay that allowed us, with generally good reproducibility at the time, to determine the IC50 for zidovudine. And we could characterize viruses from the participants in this trial as either high level or moderate or susceptible. And what you can see is that the presence of high level resistance was associated with a significant increased risk of death in people who continued to receive zidovudine. Actually, this was independent of their treatment assignment because even after controlling for assignment to didanosine, this risk persisted. Switching to didanosine reduced your risk, but there was still a relative increased risk compared to not having resistance. That's because we didn't understand there was cross resistance to didanosine as well. We could also show that presence of the combination of critical zidovudine resistance mutations, changes at codons 215 and 41, likewise had an increased risk of progression to the primary endpoint, as you can see here in the adjusted analysis, and a substantial increased risk of death, 3 to 5-fold. So that was evidence that resistance mattered and what level of resistance or which particular mutations might matter. The situation is much more complicated when you start to look at envelope. And this is an example from ACDG 5111. This was a phase 2 study of an investigational CCR5 antagonist, Vicriviroc, a drug that never made it to full approval. But what's notable are several things. First of all, here we're looking at the susceptibility curves for 3 different CCR5 antagonists, TAC779, Maraviroc, which is of course FDA approved, and Vicriviroc, for 3 different sequences obtained at the time of treatment failure from participants. And what you note is instead of seeing a shift to the right of these curves, the curves are instead flattened so that you fail to achieve 100% inhibition. And for Vicriviroc, you actually saw stimulation of replication. Now, sorting out which of these mutations is actually responsible for the phenotype is really very challenging and gets quite messy, as you can see in the panels on the right. Here, what we did was to take for one of the participants the week 28 sequence when the resistance was present and back mutate substitutions at 2 key positions. And you can see here, looking at the Vicriviroc, that these filled diamonds represent the week 28 sequence, and the open squares show what happens when the 306P mutation is reverted to the wild-type serine. And there is some restoration of the curve, but you still only are able to achieve about 50% inhibition at the highest drug concentrations achievable in the assay. by comparison, a monoclonal antibody against CCR5 had very nicely behaved sigmoid inhibition curves. Now, the way that the approach that's typically been taken to try to identify which mutations are important and what fold resistance is clinically significant has involved some very complex regression modeling, as was done for etravirine in the duet studies. And I show this slide only to indicate that these exercises are not for the faint of heart. But what the group from Tibotec did was to look at mutations that they had identified in phase 2 trials of etravirine, and then pool the data from the duet studies to look at correlations between the fold change in etravirine at study entry and the change in reduction of virus load over the course of the study. And you can see that there is a correlation, although it's a fairly rough correlation with a lot of points that fall way off of the regression line. And that can be for many reasons. So, for example, for those people who had susceptible viruses, but nevertheless failed to see much in the way of viral load reduction, perhaps that's due to non-adherence. For those who have a high fold change, but nevertheless achieve a significant reduction in virus load, perhaps that's because of the activity of boosted darunavir in their regimen. So when you try to put this all together, you can do an analysis where you explore the proportion of participants who achieved complete viral suppression by the Tullover analysis, and how that related to the fold change in IC50 for etravirine. And you can see that you don't really get much better than 70 to 77%, whereas those who have the highest level of resistance are only at about 37%. You can also do the same analysis for the mean change in virus load by category, where there's a clearer stepwise relationship. And that's been used then to try to categorize the impact of the weighted etravirine genotype score based on the impact of individual mutations on the phenotype, and then categorizing them as those that were associated with the highest response. So you could have a score of up to 2.0. Those that had an intermediate response, where only half of participants achieved viral suppression, and you could have a score up to 3.5. And those who had a reduced response of just about 38%, which was really not much different in that trial from the placebo arm, the people who did not receive etravirine. But you can see that even within each group, there's a fair bit of heterogeneity in the response. Now, for those of you who are still looking at resistance test reports, and I use one here from Monogram just for purposes of illustration, you know that this can be quite challenging to look through. And here make the point that when we ask about cutoffs for susceptibility, those shown in bold are considered to be clinical cutoffs. That is, there are data showing that viruses that have an IC50 or a fold change above this bolded value have reduced or no response. When there are two numbers, it means the lower threshold and then the upper threshold. So in between, there might be expected some intermediate response. And the values that are shown unbolded are for drugs where we really don't have any biological cutoffs. The cutoffs are based on the so-called biological cutoff from the 95% distribution of susceptibility of supposedly wild-type viruses. But that makes it really quite challenging to be able to interpret the fold change in any given person's virus. So to conclude, identifying resistance-associated mutations and defining phenotypic cutoffs for antiretroviral susceptibility is quite complex. This complexity increases dramatically when considering hypervariable targets such as envelope. Interpretive algorithms require regular updating to incorporate new drugs and viral evolution, a major problem when phenotypic tests are not ordered routinely from the clinic. And updating algorithms requires a regular flow of samples for testing. So with that, I will conclude, and although I won't be part of the discussion immediately after in this session, I hope to be able to catch up with you later in the meeting.
So next we have Jackie Reeves from Monogram LabCorp. Thank you so much. I think Dan set you up perfectly. Now you can walk us through all those reports and explain to us exactly. I know that's not what you're going to do, but we really look forward to your reminding us of all the lessons we learned from the CCR5s. Thank you so much.
Hey, thanks, Veronica. And I have some data that complements what Dan presented. So I'm going to present lessons from CCR5 antagonists that may have relevance to the development of bNAbs for HIV. So as way of outline, I'm going to present the requirement of a screening assay for the use of CCR5 antagonists, discuss the positive and negative predictive value of the profile assay in CCR5 antagonist trials, discuss how minor populations of CXCR4 using viruses were associated with treatment failure, and how enhanced detection of these can improve clinical utility. And to Dan's talk, show also examples of how reduced maximal percent inhibition can be associated with treatment failure, how complexity presents challenges for genotypic assay development. I'll touch on how we partnered for international commercialization of a tropism assay, some barriers for CCR5 antagonist uptake, and how we adapted to treatment paradigms. So Monogram supported the development and clinical use of many HIV therapeutics, including CCR5 antagonists that include Maraviroc and Vicariviroc that target co-receptor engagement. And I'll show examples primarily from these inhibitors. So to enter a cell, HIV uses a CD4 receptor and a co-receptor, typically CCR5 or CXCR4. And viruses that use CXCR4 only are 5-tropic, CXCR4 only X4. Dual-tropic viruses can use either receptor, and mixed refers to a population with 2 or more tropisms. R5 viruses predominate early in infection, and CXCR4 using viruses can develop later. And this is exemplified by data from a number of cohorts and clinical trials and treatment naive and experienced individuals. So in naives, R5 viruses are found in more than 80% of the individuals, whereas in experienced, between about 50 to 60%. So in experienced, there's a lower percent of individuals who could be candidates for CCR5 antagonists. The Maraviroc PI states that prior to initiation of treatment for HIV-1, all patients should be tested for CCR5 tropism with a highly sensitive tropism assay. So Maraviroc is recommended for patients only with CCR5 tropic virus. And it was shown in clinical trials that outgrowth of pre-existing low-level CXCR4 dual mixed tropic virus, not detected by tropism testing at screening, was associated with virologic failure. So the profile assay was used to screen individuals for entry into Maraviroc trials. A patient plasma or serum sample was used to amplify full-length HIV-Ms. These were cloned into an envelope expression vector to generate a patient M expression vector library. This library, along with the HIV luciferase reporter vector, is transfected into cells to produce pseudovirions, that are used to infect target cells that express either CCR5 or CXCR4 in the presence of co-receptor ligands as specificity controls. My screen has gone off. I don't see there. Thanks. And depending on the relative infection of these cells, we can determine whether viruses are R5, X4, or DM. We report out tropism and whether activity of a CCR5 antagonist can be anticipated. So the positive predictive value of the trophile assay is demonstrated in clinical trials with Maraviroc in Motivate 1 and 2, where populations that had greater than or equal to 3-class ARB experience and or 3-class resistance, Maraviroc demonstrated significant antiviral activity in individuals with R5 virus. And in the ACTG 5211 trial that Dan mentioned, in a population with advanced disease and high-level drug resistance, Maraviroc demonstrated significant antiviral activity, again in individuals with R5 virus. So the negative predictive value is demonstrated in 1029. So this is a population of antiviral experienced individuals that had dual and mixed tropic virus at study baseline by the trophile assay, and where addition of Maraviroc to OBT didn't provide added antiviral activity as expected in this safety study. So between 8 and 10% of treatment experienced individuals enrolled in clinical trials of Maraviroc and Maraviroc had changes in tropism between when they screened into the study with an R5 virus and at study baseline when they had DM virus. And this reflects the presence of DM virus around the assay detection limit. And these patients had reduced virological responses compared to those that had R5 virus at screening baseline. So an enhanced sensitivity trophile assay was developed to increase minor CXCR4 using virus detection, with the aim of further optimizing the selection of patients who may benefit from CCR5 inhibitor therapy. And clinical trial reanalysis was performed to evaluate if the enhanced assay would better predict virologic outcomes. And so from an analysis of sensitivity to detect minor variants, we found that X4 variant detection by the original assay was 100% if the variant was present at 10% of a mixture. And within the enhanced assay, it was 30-fold improved with 100% detection of a 0.3% mixture. The MERIT study enrolled treatment-naive individuals with R5 virus who were randomized to receive either rifavirin or Maraviroc with Combivir. And based on the pre-planned statistical analysis, Maraviroc did not meet the non-inferiority margin that was set at the less than 50 copies endpoint. 58 subjects failed the trial due to lack of viral efficacy. 0 of 15 of the failures in the rifavirin arm were not associated with a switch with tropism. Almost half of those in the Maraviroc arm were associated with a switch in tropism. So enhanced sensitivity tropism testing was performed on the stored residual vectors from the trial of all patients who had R5 virus by the original assay. The enhanced assay was able to reclassify tropism to DM at screening in 15% of patients, and the non-inferiority criteria was met when patients with DM virus were excluded from study analysis. And this retrospective analysis supported the naive indication from Maraviroc. In the 5211 trial, 118 subjects with R5 virus by trophile and in a failing ART regimen were randomized to receive either placebo or Maraviroc for 14 days prior to addition of optimized ART. In this study, 12 of those individuals with R virus at screen had DM virus at study entry with the original assay, and this virus pretended an attenuated response compared to that that had R5 at screen and entry. An additional 18 individuals who had R5 virus at screen and entry had CXCR4 using virus detected on study by the original assay. So the enhanced assay was used to reevaluate tropism in these individuals. So of the 12 with DM virus at entry, 58% were reclassified as having DM virus by the enhanced assay. Of the 18 with CXCR4 use detected on study, 50% were reclassified with DM at entry. And of 88 with no CXCR4 using virus detected, only 10% were reclassified with DM virus. So in total, the enhanced assay reclassified 25 individuals with DM virus at screen. Of those 25, 15 received Vicroviroc, and 12 of the 15 had early emergence of CXCR4 using virus as detected by the original assay. Greater viral load reductions were observed at day 14 and also week 24 as shown here in subjects that had R5 virus at screen and entry compared to those that had DM virus at screen. Now, as Dan mentioned, as well as outgrowth and minor variants, you can see two profiles of entry inhibitor resistance. So competitive inhibitors such as enfuvirtide resistance is observed by an increase in IC50. For non-competitive inhibitors such as Maraviroc and Vicroviroc, resistance can be manifested as a reduction in maximal percent inhibition. It can also lead to enhancement of infection, as Dan showed in his presentation. So for enfuvirtide resistance here at baseline compared to week 16, when resistance developed, we can see a 250-fold shift in the IC50. And with Vicroviroc, evolution of resistance can be manifested as a progressive reduction in the maximum percent inhibition for continued Vicroviroc treatment. The sequence diversity of HIV-M within and between individuals can present challenges for genotypic prediction of tropism. The V3 loops are primary determinant of tropism, but regions in GP120 and also in GP41 can influence tropism, and this limits the sensitivity of genotypic algorithms. And when compared to phenotyping, genotypic methods based on V3 sequence have relatively high specificity but low sensitivity for predicting CXCR4 use. And this is exemplified with this data set. So dual tropic viruses that have relatively low CXCR4 use compared to R5 and are termed dual R here are difficult to detect by genotypic algorithms, the V3 loops, relatively similar to R5s. In contrast, dual X viruses that have relatively efficient use of CXCR4 can be more readily detected in some instances by these algorithms. A phenotypic assay is preferred to determine HIV co-receptor tropism according to DHS guidelines, and this is because genotypic assays may not be sufficiently robust to completely rule out the presence of X4 or DM virus. However, there is moderate recommendations for the use of a genotypic assay as an alternative test to predict co-receptor usage. So Pfizer partnered with Monogram throughout the clinical development of Maraviroc through to approval and commercialization. The TROPIL assay was used for clinical trial screening, and we collaborated on the reanalysis of the MERIT data, and we partnered to facilitate an international expanded access program and for international access to the TROPIL assay upon Maraviroc approval and availability throughout the US and North and South America. And we had a sponsored testing program initially available for no cost ex-US access to TROPIL. Barriers to the use of CCR5 antagonists include that a subset of individuals only are eligible for treatment. There's a requirement for the selection assay with associated costs and turnaround time. Maraviroc had BID dosing, And the timing of approval from Ravarot coincided with that for valtegravir that had favorable properties, QD dosing, it was tolerable, and no selection assay was required. Bicravirac also went into phase 3 development but didn't progress further. So at the time of the phase 3 trial, patients had access to new drugs for OBT that was effective at controlling the virus. Now, selection assays were originally for viremic individuals only and had a viral load requirement of at least 1,000. And the ability to switch or to incorporate Maraviroc into a suppressive treatment regimen being precluded by the viral load requirement. So we developed a DNA version of the Trofile assay that isolated HIV from infected cells to generate the resistance vector library. Assay sensitivity of these DNA-based assays is determined by the number of copies of HIV that's in a sample, and most individuals have less than 100 copies of the target DNA in a test aliquot. So for DNA assays, there are sampling limitations, while conventional assays based on plasma RNA have a minimum viral load requirement that ensures adequate representation. The newer assays based on sampling of cell-associated DNA are more vulnerable to sampling bias due to limitations in sampling size. So the tropism of virus sample can also be dependent upon the compartment that the virus is found in. We performed a study here that demonstrates this, where tropism was evaluated in 46 paired CSF and plasma samples. In 90% of the cases, the tropism was concordant in the plasma and CSF. But in 10%, there was some discordance with either DM virus in plasma, R5 in CSF, or vice versa. So to end, from lessons from CCR5 antagonists that might have relevance to bNAb development, screening assays were required for optimal efficacy of CCR5 antagonists. Collaborations between pharma, academic, and diagnostic partners facilitated optimization of the co-receptor tropism assay to improve clinical utility of CCR5 antagonists. Minor populations of CXCR4 variants and reduced maximal % inhibition can be associated with treatment failure, and there's also potential for virus to outgrow from other compartments with tropism differences. Envelope complexities prevent challenges for genotypic assays, benefit from pharma diagnostic partnerships throughout drug assay development and commercialization were obtained. And to minimize barriers to drug uptake, we've partnered to facilitate accredited assay access and coverage, but the solutions in US and EU may vary. And timing to treatment needs to be a viable competitive option relative to others. I'd like to end by acknowledging colleagues at Monogram and many academic collaborators at different institutions and within different pharma companies. Okay, thank you. Speakers up. There we go, thanks. And Sean?
Just for share.
Okay. Great, thank you. And thank you so much for that great overview. Jackie, those were, that was a lot of work.
to develop the assay and fine tune it. I remember well those days and the discussions about the accessibility and how to manage all of that. So congrats again to you and your team for everything you were able to do there. But let me introduce our additional panelists, and Dan will join us when he's able to come down after selecting the fellows for their position. So we have Simon Collins next to me. Simon, would you just like to say hello and introduce yourself briefly?
Yeah, sure. Thanks, Veronica. So my name is Simon Collins. I'm an HIV positive treatment advocate with an organisation called iBase, which is based in London in the UK. But I'm also connected to various community networks like the European AIDS Treatment Group, the UK CAB, and ATAC, which is a similar organisation in the US. I've always been very interested in the potential for bNAbs and the sort of particularly the sensitivity of the tests to identify sensitivity at baseline.
Great. And we have Sean Collins.
Hi, everybody. I'm Sean Collins. I work with Gilead Sciences. I'm on the clinical development side, and I lead our lenacapavir plus broadly neutralizing antibody clinical treatment program.
Great. And Benjamin Lawrence.
Hi, good morning. Ben Lorenz, one of the medical officers with the Division of Antivirals at the FDA. We're within CEDAR, so concentrating more on a little bit of a clinical perspective in the development of antivirals. So,
Great. So, right. So one of the topics we wanted to get into a little bit was the sort of statistical versus clinical resistance. And Dan did a nice job of reminding us early on in the days when we weren't measuring viral load during the ACTG 116, 117 days, that you could actually see the impact on clinical progression and death if people's viruses were resistant to zidovudine. And we later on showed that that also happened with AZT3 double resistant viruses. Now, of course, we measure the clinical impact on viral load, and we can, of course, hopefully have other regimen that we can put in to resuppress the viral load. But let me just put that question out there before we get into other questions on this panel. Maybe, Joe, you could comment a little bit on that question of statistical versus, and again, thinking about the lessons we've learned and thinking forward to broadly neutralizing antibodies. How will we, and this is sort of a question for later, but just to think ahead, how will we get at that clinical significance of lack of suppression with bNAbs?
I actually think we have a pretty big advantage with bNAbs that was kind of different than small molecules, because in the population of people, there's probably a range of susceptibility to an individual bNAb. For example, in a population of people, the range of susceptibility to an integrase inhibitor is like 100%. Everybody goes down. But in bNAbs, there's clearly a range. So the way we're studying these are looking at essentially single therapies, single drug therapy over some short period of time, whether it's 10 days or 14 days. Because there's a range of susceptibility, you can actually look at an association between susceptibility and at least that outcome, change in viral load, whether that will be, what correlates with maintaining suppression or not. But I think that as opposed to, taking, 100 or 1,000 viruses from a population and doing a bell-shaped curve and just making your definition based on statistics, we actually will have some relationships that can be created. And I know that Peter actually presented something like that I think last year or 2 years ago at Croy. So I think that's an advantage that now whether that is going to predict outcome in combination, whether that's going to predict sustaining suppression, I don't know. But I think that gives us a bit of a leg up on just looking at, you know, a broad population of what we think are wild type viruses and making a statistical cutoff based on a bell-shaped curve, which I think was what, you know, Monogram was forced to do for drugs like Nalfinavir that you know, Dan pointed out. They just had a you know, make it. So that's my thought.
Great, thank you. Thank you very much. Any comments on Joe's comments in terms of, you know, as we proceed to talk further about sort of delineating the role of diagnostics and bNAbs? But I think that makes a lot of sense in terms of it. Dan's review also reminds us how far we've come, right? That we're not looking at a twofold increase of disease progression and death anymore, which is, you know, those studies were done not so long ago. But let me then go along the panel here and kind of trying to take the lessons from molecule antivirals and entry inhibitors to what we might expect in bNAbs in terms of diagnostics and the challenges. Let me start with you, Sean. And what are your thoughts, your takeaways from these two overview talks, and where do you see the key questions lies for us to move forward here?
I think one of the main challenges that I see is the clinical correlation side. So as we look back historically on how we've made some of these cutoffs, we can make an in vitro cutoff, but we are also looking for that in vivo clinical correlation. I think that part may be more challenging where we are with bNAbs now and sorting that out back directly to the in vitro sensitivity data. That's where I think the biggest challenge is. The other piece that I think is kind of like a very interesting piece of how it will come together, to Joe's point about people who are viremic and looking at that and what's the role and how do these tests, how can they perform in people who have plasma virus versus individuals who are fully suppressed, and we're looking at doing proviral DNA and what's the relationship and how well can we characterize that.
Great. And from a regulator's perspective, Ben, your initial thoughts?
Yeah, I mean, the key is clinical correlation with activity. I don't think we can necessarily say that there's a direct correlation with how we developed and looked at Maraviroc per se and bNAbs, which are more direct acting. And so, you know, it depends on what mechanism of action we're talking about and how deterministic we expect the diagnostic to be. But from the perspective of core, that I think we encourage use of the diagnostic during clinical development of whatever study drug we're talking about. If we're talking about a bNAb, then of course I think we encourage it, but would require it for approval. So
great, thank you. And Simon, again, your initial comments. You've lived through that CCR5 and a period and challenges to access, but what are your initial thoughts as a very experienced, and I mean experienced in all ways, patient who has seen all of these treatments come up, and what are your thoughts as you're considering bNAbs as a development for therapeutic indication?
So very interesting meeting. Yeah, and we've been on drug resistance panels years ago, maybe 20 years ago. Yeah. And so all this sort of the same science coming back now, the main concern from an advocacy perspective is development of drug resistance by use of suboptimal regimens where HIV progresses and people lose the option of this fantastic potential because they're using suboptimal bNAbs or bNAbs that where part of the combination, we should be talking about combinations just like with HIV drugs, where part of that is not going to be sensitive at baseline. And so the experience about Maraviroc really comes from, I didn't think that the testing pre-use was never an issue in itself, but certainly access to phenotypic testing is a big problem unless you live in the US and you can fast track your samples there. And so Europe overcame the inability to access phenotypic testing by developing genotypic equivalents, which were pretty sensitive. You know, they worked in clinical practice. No one sent samples from Europe to the US in order to test them, but the development of genotypic assays was was pretty effective. And so we need something similar. And the current block at the moment, there was a comparative analysis of phenotypic versus genotypic bNAb sensitivity tests that probably a couple of years ago. And although the conclusion was that there's pretty good overlap, as an advocate, I didn't read that. I think maybe 10 or 20% of people were inaccurately identified as either being responders when they didn't respond, or being non-responders and they lost the opportunity to use bNAbs as treatments. And I'm pretty sure that some of the responders in the early bNAb trials, when they were subsequently tested, and they were later tested for sensitivity, would have been excluded from the studies because they were some of the best responders. So key points really, we should be doing this in combination. The combinations may cover some of the lack of sensitivity and specificity of the tests themselves. And please think about global access. And I think that even though FDA has approved the phenotypic test, I just hear too many people saying it's not sensitive enough. And one other point, is that we need to be doing this now. We can't wait until bNAbs are approved, even though they're tentative and speculative, and we're hoping to see great opportunities for use in treatment and prevention. We really need to be working on the assays at the same time.
Right. And Jackie, back to you, in terms of the comments you have heard, and also maybe thinking, you know, the field has advanced so much since you know, the beginning with AZT and the initial development of the phenotypic and genotypic resistance assays, but also the whole field of diagnostic development and the challenges for approval. So first of all, the acceptance of an assay to be part of a clinical trial, to have that key data be acceptable, and then for the assay approval for clinical use through CDRH, and the challenges there in terms of, you know, all the different parameters, and of course the reimbursement scene has become much more challenging as well. So how do you see this field progressing in terms of all of those challenges, and especially also, you know, we're focusing on the US, we don't really want to get into the IVDR and the European situation here, but as an assay developer, how do you see that sort of the progression and changes in diagnostic development over the decades?
Yeah, so there's definitely going to be upcoming challenges with the new regulations in Europe. For Maraviroc, we partnered with 6 labs in the EU that basically ship samples to us for testing, and within I think an average of 22 days we would get results back to EU, and that was a partnership that was set up with Pfizer, and testing was sponsored by Pfizer. So we had a mechanism to make the test accessible. Now with the new regulations in Europe, there's going to be much more challenges getting a test in the US that could be accessible in Europe. for clinical trials, I think it's well accepted that testing is done to support the selection of patients, to analyze the outcome, to look at resistance and to try and find correlates of susceptibility. And but with bNAbs, as was stated, that there's a wide range of variety and susceptibility to bNAbs. A lot of them, there's more than 3 log difference in susceptibility. Some people that have got highly resistant virus can be successfully treated if you've got the right background regimen. Others with highly resistant virus can rebound pretty quickly, and vice versa. Some individuals that look like they have sensitive virus can be successfully treated and others rebound. So there's definitely challenges trying to select individuals, but I think the collaboration between the academics and the companies developing the drugs to try and optimize selection, to dial in the right threshold, to look at correlates of response and non-response is important. Some of the early testing we've done for different trials and for some retrospective analyses that reduced maximum percent inhibition seem to be really important. If you've got a subpopulation of virus that's resistant to start with, that seems to particularly portend resistance. And that's in contrast to iberalizumab. So iberalizumab is CD4 mAb. There you can have pre-existing plateaus and reduce maximum percent inhibition and some individuals can be successfully treated then. And there, a growth of that plateau, so a decrease in the maximum percent inhibition is correlated with resistance. So I think it's trying to dial in the right threshold. And in the context of active regimens, then trying to find clerical correlates is harder.
Right. So, so then in terms of moving forward and sort of thinking about the lessons from the past were because of many different factors, the difference in the small molecules and entry inhibitors and bNAbs, and also other monoclonal antibodies like you. I'm glad you raised the in your talk and just now also talking about the anti-CD4s. But in terms of then moving that forward, I remember those early days of drug resistance, the meetings that we had, the collaborations, the number of samples you guys and other labs received, and the number of genotypes that were done. Bob Schaefer's amazing database that was accessible to people, where you could enter the mutations that your patient had, and you could get a readout on the likely response to that. How do we build that these days? And also, I think in terms of the challenges of the analysis, again, that's another area. So antivirals, the antiviral therapeutics, the clinical impact on people has progressed so much. The diagnostic development field has progressed so much too, but so have analytics, right? And machine learning and how we actually learn, from very complex data. So how do we move to that place where we have the, let's call it the Stanford, just as a, placeholder, that kind of a database that we could probe and look at and really all learn together as different companies are developing tests. And you are, you have your susceptibility neutralizing assay. Other people are developing genotypic assays as well. So how do we move towards that kind of data richness that we can actually learn from? What needs to happen from there? And just leaving this wide open for discussion. How do you think that would that be helpful? Something like that, would that be helpful for you, Ben, and your colleagues, to really be able to dig into data and really, try and get at some of these correlations between mutations and viral load and neutralization, et cetera?
Yeah, it might be a little helpful. I think, it might be helpful from a population perspective as well to understand, where geographic
Mhm.
distributions lie. I think one consideration might be is, if you conduct a trial abroad and, how applicable it would be to the US and vice versa. So, from a population perspective, it might be helpful, if there's differences in clade activity or something like that.
Mhm.
So, I think that might be potential consideration.
Databases.
Yeah, please.
Well, I mean, where you've got currently low data, then shared databases is collaborations and shared databases will be the only way to get sufficient power to understand the implications of different mutations. And
Sure.
what the point, I guess, on clade is also really important.
Mhm.
When you've had large studies that use antibodies that maybe aren't susceptible to the clades in the countries they're being used, we need to sharpen up on that a little.
I would kind of echo the need for testing. If we're gonna be able to build databases, we need a whole lot more. And if we're talking about AI, we're talking about orders of magnitude more data than we have now in terms of pheno correlations. I would also echo the need for enhanced understanding of geographic specificity and sensitivity, because although it's really important for us to think about what the development of broadly neutralizing antibodies means globally and what access can mean globally, it's also important to reflect that there are likely to be large geographic areas that certain antibodies are the wrong thing for. And so we need to make sure that there's specificity in our planning there. And then the other thing I think I would ask as a question, more than I have the right answer to, is how much do we feel we can tolerate individuals failing these regimens that are in experimental development? Because when we're utilizing sensitivity screening, and I can at least speak to the way that I've utilized it, we're really trying to select individuals who we think have the greatest chance of success on that regimen, recognizing that there's some uncertainty. But in order to look at clinical correlations across the breadth of that assay, you might choose different characteristics, and that might increase the likelihood of clinical failure, which is something in general we try to avoid. And I guess I'm curious what others think about that dichotomy.
Sort of that in vivo dose response curve, for example, in terms of not dose response, but susceptibility. Yeah.
Yeah, as Simon referred to, we know that certain people who have responded, if I can think of some of the studies that Rockefeller did, some of the people who were their longest term responders were people who within one or another part of their susceptibility testing would have not been predicted to respond. So how do we explore that? Do we explore that, and what's our tolerance for individuals who may not respond? Not for the individuals, but for that risk that we take on.
Okay, we have Joe, Aaron
Yeah,
here,
but-
and really want to also encourage our people participating online to send in your comments and questions.
There does look like there's a chat thing there. I can't, I don't know if you can see it, Veronica.
Yeah,
but,
so far we just had a comment back on the previous comment,
Oh.
or am I missing something, Logan? No, okay. good.
So, yeah, I just want to be respectful of the people. So I have a couple of questions. One is to Jackie, and with the mechanism for neutralizing antibody, do you anticipate that you'll see a maximum % inhibition issue, or is it going to be more like Infevertide and antiretro small molecules in terms of what you might predict, whether it's using plasma or DNA? And maybe I'll let you answer. Then I have a comment about a separate comment about what Sean was talking about.
Yeah, so we see both with bNAbs. So some bNAbs resistance manifests as a shift in IC50 and others an increase in decrease in maximum percent inhibition. So we see both profiles of resistance and some pre-existing plateaus can correlate quite well with reduced treatment. So I think MPI is particularly important as well as
and does it matter what the target was, like CD4 binding or V3 loop or whatever? Or you just see it with all the different targets?
So I think in studies with 10-10-74, that's maybe more prone to have pre-existing reductions in maximum percent inhibition that correlates with reduced
Fascinating.
reduced efficacy.
I didn't realize that. That's really helpful. And then the other, I think, and this gets back to the very first comment that we got, that the one issue is these, antibodies are so incredibly safe that you can give more of them, right? So you have to like decide, you're going to give 10 milligrams per kilo, 20, 30, you can give more. I mean that, but then there's of course a cost issue and an administration issue and all those other things. But they may work if you give more. And I know that's a decision that people who develop these antibodies have to make. That may not solve the percent maximum inhibition issue.
So yeah, with shifts in IC50, you can add more drug and
right
cover that resistance off. But with MPI, you add more drug and the
fascinating
PMI can stay the same.
Thank you.
Great. And also in terms of the distribution of susceptibility or ability to be neutralized, how does that relate to time since infection?
So we see, actually, we haven't looked at susceptibility to the newer bNAbs across the infection range. So some early studies looked at bNAbs in different stages of infection, at least naturally antibodies development over time. That the virus escapes the antibody and there's evolution. I don't know of any data showing whether there's a difference in susceptibility to bNAbs, the current generations of bNAbs in different populations, depending on how long they've been infected. But I mean, it's possible that as the virus evolves and there's more complexity, there may be more of a range of susceptibility within an individual.
Yeah, just thinking, we have a comment.
Yeah. Hi, if I could comment on that. We've looked at some of our,
Could you just introduce?
oh, I'm sorry.
yourself so people online know who's talking.
Hi, I'm Nicole Doria-Rose. I'm at the Vaccine Research Center at the NIH. And so we've looked at some of these broadly neutralizing antibodies and on large panels where we have viruses, where we have, not across one person's course of infection, but some of the viruses in our panel are CCR5 using and some are DM, et cetera. And we have not really seen correlation in sensitivity to these antibodies dependent on CCR5 versus CXCR4 usage. That doesn't mean it's not there. We just haven't seen it in a data set of 200. We haven't seen that.
Right. Because that might be a difference again in the application to cure research where you would be targeting people with much more recent infection and also in prevention of mother to child, right, in terms of the clonal expansion in the infant, if the infant were infected. Good. So Dan, welcome back. You gave a very good talk. Nobody fell asleep, but no, it was really excellent to have that review and going all the way back to those early studies and demonstrating that there was clinical impact of drug resistance, which in the early days we measured by disease progression. And in terms of the, we've kind of talked a little bit about, obviously therapeutics have advanced incredibly, so has diagnostic development, and also the increased maybe stringency and overview of what happens, for things to get reimbursed these days, for example, is a much bigger hurdle than it was, I think, at the beginning when people just accepted that there was clinical utility to HIV drug resistance testing. Not saying that it wasn't without its challenges, but also then in terms of analytics and our approaches, I think we've always used some form of machine learning, even if very primitive, but advances in analytics and applications of correlations, et cetera. So thinking about how complex this topic is with the V3 loop, with people already being on treatment and contributions of other agents, how do we tease all of this out? In your talk, you also mentioned the importance of data flow. So we touched a little bit about on that in terms of where is the data coming to? How do we replicate what Bob Schaefer did at Stanford, so that we can all, because that was an amazing effort there. So that's kind of been the discussion here this morning so far. And kind of trying to stay within the theme of what have we learned from those other experiences in terms of how we move forward. So in terms then of the, I think the lessons we have learned and from CCR5 and the earlier drug resistance studies is that, first of all, it takes a village, and village writ large, right? No single sponsor can really figure this out on their own, and no single company can really work on this on their own. So if we're going to be serious about developing this field, we need to think of that. That needs to be factored in from the very beginning in terms of what data we're gathering, what we're learning, and opportunities for collaboration across companies and sponsors and NIH-funded research and other settings. And I think we can agree all that it's not easy. What you guys did at Monogram was not an easy task, and what other people did in Europe with the genotypic phenotypic correlation. I mean, none of that was easy, and so I think if we think ahead to a day when bNAbs will be available for therapeutics, we need to really factor in sort of that level of collaboration moving forward right away. And I think the other lesson is is that having a required companion diagnostic in a field where there are a lot of other opportunities for treatment, unless there's a very specific indication for bNAb along some of the lines that Joe was talking about, that really does make it very difficult, from an implementation perspective. And this maybe does get back to your comment also, Sharon, and that, what is the place of this and the global health context, and what do we need to think about now in terms of assuring global health access. But a companion diagnostic, when you have so many other treatment options that don't require one, is not a welcome part of the market. But at the same time, we need to walk into this with eyes open and really test that scenario. Will we always need to test the susceptibility or the epitopes of the virus that a patient has? So I think that clearly is one of the lessons there as well. So if you were advising Simon, a company, on how to move forward here, what would be your 2 or 3 key messages, recommendations to a sponsor?
Well, I think the main lesson we learned from the history of drug resistance for antiretrovirals was the need for combinations.
Mhm.
And what you want to do is you want to avoid resistance, because if you develop resistance, that's like HIV disease progression. It's cutting out your future options. So whether they're tri-specific antibodies or the combinations of antibodies, let's not go through monotherapy, dual therapy, and accumulating drug resistance. I had a question actually, whether the mechanism, whether the target that the antibodies use pose different challenges to the sensitivity testing. You know, whether they're intrinsically easier if you're using V3 or V2 loops, where you have overlap with experience from Maraviroc. You know, if you're using other more distant targets or more difficult targets, is that more susceptible, a more difficult challenge? Sorry.
Yeah.
And in terms of advice for companies, I don't know. You're the expert of this. I mean, I'd be frustrated to see people not accurately identified at baseline who develop resistance unnecessarily. I'm just as concerned for people who are really keen to go into these studies, but actually they're then disappointed because they fail a sensitivity test, which later on down the line might not have been necessary. So working together, I guess, is the most.
So maybe we'll go to the question to Jackie about the target, the specific target on the envelope.
Yeah, so the target, so whether antibodies target the B3 loop, CD4 binding site, or GP41, or wherever on the envelope, the assays are amenable to test equivalently the susceptibility. So there's no challenge to doing the neutralization assays depending on the specificity of the antibody.
Thanks.
And maybe Sean, maybe comment on Simon's comment. He was looking at you.
I think what he wants is what we want.
Yeah.
From the development standpoint, we want if we are using a test, especially for a screening test to use a regimen, we want it to perform well on both sides of the spectrum, both to include people who are sensitive and to exclude those who are resistant. We want to do both of those jobs as accurately as possible. But I agree, it is currently a challenge to say that we're doing that with currently available tests, and it requires additional work and additional development to try to improve along those lines.
Okay, Richard.
Yeah, I'm not really sure if this is a valid question, but given the variability of these targets, is there a greater risk that the resistant variant is kind of elsewhere in the body from where you've sampled? Like if people looked at paired tissue and blood samples and compared the profiles to see if they're consistent or different?
So, so the question here is, in case it didn't come through for those online, viruses and different tissues and paired studies in terms of neutralizing assays. And I know definitely you had a slide with CSF, but that was for CCR5, of course, yeah.
Yeah, so for co-receptor antagonists, CCR5 inhibitors, we've definitely seen differences in compartments, and some early studies have shown that viruses taken from different reservoirs can have different susceptibility to entry inhibitors more broadly. So yeah, that could be a challenge to efficacy.
Good, Jörg?
There are actually some studies, small studies, where they looked at sequences from virus from the CNS, and then the pseudoviruses tested those. I mean, because we know there's compartmentalization, in the CNS of viruses, and then they tested if they are susceptible to bNAbs as well, and there are differences. Which, that's exactly the question. Where do we sample? How often do we sample? Is a single blood draw enough to give us the whole reservoir kind of diversity with all the different envelopes that could potentially come up? And then bNAbs obviously don't go to the CNS. So if you treat the peripheral virus, you don't suppress maybe virus that relapses in the CNS and then comes over and it's resistant that we didn't detect before. We didn't sample I mean, I think these are all still some of these testing complications, I guess we're going to talk about later.
Right, that will all, we're just setting the stage for the rest of the day. Any other comments from Vani, please?
Vani Vanapagari, I'm the epidemiologist from Weave Healthcare. So you know where this is going. The population level distribution of the sensitivity pieces. You know, taking the examples from G6PD deficiency or HLA-B5701, it took a long time for all the stakeholders to build those databases and then come up with the prevalence of this, the various polymorphisms. So what are we doing with respect to the bNAbs sensitivity assay data by geography? Ben, Sean, Simon, you all alluded to the fact that that's going to be something of importance, whether a particular geographic area will benefit from this treatment regimens or not. So right now we do have some data from the smaller early studies. Is there an effort to publish that data? Jackie, I'm looking at you to see that would be the starting point. And then as studies continue, those can keep adding to the database and what we know about the geographic distribution.
Yeah, we've done studies with some of the early broadly neutralizing antibodies to look at neutralization of clades from a different viral clades, and you can see quite large differences for some of the antibodies. So, you know, clade Bs might be effectively neutralized, and some non-B viruses may be very poorly neutralized. And so I think, yeah, evaluating breadth and neutralization among different virus clades can be particularly important. And I think, Dan, you've done some studies in patients with non-B virus that may help address the question as well.
Yeah, no, I was just going to make 3 quick points. This is Dan Kuritzkis from Boston. One, I think we have to differentiate between whether resistance or susceptibility testing is going to be required or merely desired. You know, with new classes of antiretrovirals, typically we haven't worried about resistance because with only a few minor exceptions, there's been general universal susceptibility. That's clearly not the case with bNAbs, nor is it the case that even with combinations of bNAbs that the virus will be susceptible, that all viruses will be susceptible to each bNAb, which is a key distinction between combination small molecule therapy and combination bNAb therapy. The second is that it becomes essential then to be able to define a phenotypic threshold for susceptibility. And for all the reasons that have been talked about, it's very complex when you're dealing with envelope. And then the third is if you can agree on what is defined as susceptible, you then have the additional challenge of can you substitute sequence information for phenotypic testing, which is enormously more complex for envelope. There may be some differences to Simon's point when you're talking about glycan-directed antibodies predicting resistance because you know about the canonical glycosylation sites, but that doesn't mean you can as easily predict susceptibility.
Right, right. Very well summarized. And I think with again, with drug resistance, even for the drugs that were developed specifically like Tipranavir, for example, for protease-resistant viruses, I don't know that resistance testing was ever required. It was obviously you wouldn't give that drug to somebody who didn't need it, but it was not a companion diagnostic in those days. Please.
Yeah, so this is Yunda Huang from HIV Vaccine Trials Network. I just want to make a comment about the database for bNAb susceptibility. We've been using the Los Alamos CatNab dataset. I think it stands for CAT, compile, analyze, tally, NAB panel database. So basically over there, I don't, at least across geographical regions, you know, there's a diversity of viruses. I'm not sure about the within host evolution pools of viruses, but there are, you know, panels of acute viruses or chronic infected viruses. So you see a little bit, you know, range of different types of viruses over there. So that's a pretty rich data set, and I think they have uploads from companies and from research groups and across the globe for a pretty good collection of bNAbs over there. The other comment about basically measuring susceptibility in vivo. We have a little bit data kind of from the AMP trial, which I can speak about in the afternoon.
Okay,
Just a
great.
little talking.
Thank you so much. And again, to the people online, please do send in your comments and questions. Joe has one more.
One last quick one. This is for Simon's concern about, whether people who select for resistance will then not be able to use that bNAb in the future. In people who have a mixture, and when the antibodies not present and you select out, one with decreased susceptibility, when that antibody is removed, will it end up being, back to where it was? In other words, I mean, I don't know whether resistance will be the same thing, that where there's, kind of sequential modification like we see with protease inhibitors or integrase inhibitors, where there actually is selection of new mutations as opposed to selecting of a pre-existing envelope that then just goes back to where it was when the antibodies removed. So I don't know that the risk is the same.
So do you mean you're not worried about archived resistance?
Well, I'm wondering about, because it's my idea is already archived, right? It's already in there, right? It's pre-existing, and maybe I don't know. I'm not that, there are other people here that are more expert than me about that, and so how impactful is it if you actually select for a variant that pre-existed for that individual? I don't know. I'm just,
So I thought it developed at suboptimal drug levels. So as the tail folded off, that's when.
Oh, well, not necessarily, right? Some people just don't respond, right? There's a population that just doesn't respond, right? So it's different as opposed to a selection of kind of mutations that only exist in the presence of the antibody. I don't know. I'm just thinking about whether the consequences for an individual are the same or not. I don't know. I was just raising that as something for people that think about this more often to really, because I don't think it's quite the same thing.
Drug pressure and evolutionary pressure.
Right. As opposed to a pre-existing variant at a very low level that quickly rises because of the selective pressure that's applied. So because envelope is so diverse, right? As opposed to some of the enzymatic targets that we've had in the past. So it's just a question. It's not. I'm not saying it is or it isn't. I'm just raising it.
Right.
We need more data.
Yes. So
and we have so few people that have been experienced to these compounds.
Right.
So that's why the collaborative aspect of pooling the data.
And I think Sean's point was that if we screen out, if we only screen in the people that are very susceptible, you'll never learn anything about those people that are not as susceptible. Right. So I think that's the conundrum, right? That I think that's what you were getting at, Sean, correct or no?
Yeah, I think it'll be harder to have confidence in calling something your cutoff, whatever criteria you're using. Because
you need a curve.
Yeah, you've never tested anybody above that cutoff, so to speak.
Yeah, Peter Leone from ViiV. That's the problem in some way. So when you have a broadly neutralizing antibody, you have very few people that are truly insensitive. And so screening becomes this issue. That cutoff that's used currently is an arbitrary cutoff. The challenge is trying to push that threshold. I know we're going to wind up looking at that, but that's a really difficult thing to do because the N is small frequently for these really broad antibodies. So even getting a statistical analysis that's significant enough to actually say anything becomes the challenge, and we want it to be safe. And I think Joe's point about what's the clinical outcome from having someone who develops a change in sensitivity, I don't think we really know yet. It would be worth going back and looking at some of these populations that have failed, either in monotherapy or in some of the clinical trials. But again, the numbers are small because we're building backwards. From a pharma standpoint, you want to be safe. So you select out populations that are less likely to fail, which is sort of in some ways creating a problem, trying to answer the question here about how far can you go and how much coverage can you get. So I think it's going to take a lot of collaboration. It comes back to looking at some of these profiles in terms of sensitivity that we can't generate as a pharma company. Like, we cannot go into places in which I know we'll have discussions, the IVDR is in place. So you go, okay, if you're looking only in the US, you can't answer the question about what's the coverage in other places. It becomes a circular argument. So there has to be some collaboration to try to look at much broader bases. We are looking at CatNab for our information for coverage. That's not going to be sufficient, especially if we're trying to answer the question of how much coverage do you need? I think Joe's point about it, and I'll bring it up when we talk about it, because of the safety profile, you can probably cover almost all of these folks if you give enough bNAb. That's not feasible from a development standpoint, but that's sort of where we're at from a personalized medicine standpoint. We could probably, in the case of what we're looking at, cover 97% of people if you just gave enough. That's not real world possibility. So again, I think we're going to need some collaboration to look at a much broader aspect of moving the field forward. It's a statement and no question, I guess.
Well received. Tim?
Hi, Tim Wilkin from Cornell. Just to encourage people to have perhaps a little bit broader inclusion criteria, because as we were reminded about CCR5 antagonists, as the testing evolved, as we got more understanding and better tests, we were able to go back and look at the clinical trial results through the lens of the new testing. And it sounded like that data was accepted by the FDA. So it could be that perhaps we don't have the best test now, or we don't really understand how to use the test. But if you have broader inclusion criteria, you will have a rich data set to go back once you have defined the test. Both of these things need to happen at the same time. This may be getting towards the afternoon. This all makes sense if you're giving an all bNAb regimen. It's a little bit disconcerting if you're, developing it with lenacapavir. I would say that in cure studies and other things, we don't really think about resistance to bNAbs as a clinical harm. I think the greater clinical harm comes from virologic failure and the possibility of onward transmission or inflammation or adverse clinical events for the person, not so much from the resistance itself. At least that's how we tend to think about it in the research community.
Good, thank you. Well, I think we are, there's a new question. Oh, great. Great discussion. This is from Christine Lampkin. Can Ben comment on the expected regulatory requirement for the assay as a companion diagnostic? And I'm not quite sure if from the division.
Yeah, that's out of the scope of our division. I'd have to defer to device reviewers on that. But, I think from the point of view of, the requirement for approval, we, again, like to encourage that those devices or diagnostics be, developed in parallel or in concert with those drugs. And to the point that was just raised, some of these data can be, kind of retrospectively applied and used.
Right. So, and please go ahead.
Hi, I'm Julie Lathrop from CBER
Sorry.
sorry. I'm Julie Lathrop from CBER from the division that does regulate these tests. So we can talk about that this afternoon on those
Wonderful.
panels.
Yeah, thank you so much. And it's not always CDRH. In this case, it's CBER. So that's always interesting too, in terms of that. So I think we can, one more comment.
One final comment, Veronica, and it has to do with what Peter was saying from the pharma perspective, perhaps to build a little on what he said. And this is the same philosophical issue we faced about a year ago in the same room when we were discussing long acting. And the fact that the people who may need these therapies most, or certainly in a highly unmet need kind of way, are not necessarily the patient populations that we would normally enroll into our clinical trials, just because there are challenges, be it the virus, be it the therapeutics, be it the patient population. And going back to this of playing it safe and not going above certain thresholds of susceptibility, yes, that's what pharma is going to do, right? Because what we need to be able to do is in a relatively cost-effective, time-effective way, bring new therapies into the clinic. And pharma is not going to take certain risks. So we go back to what we discussed a year ago about trying to create perhaps a second pathway of being in a safe space, to use the term that was used earlier, where we could test these things without there being a negative consequence for the main development program that would put at risk the licensing of a drug, right? We would be willing to take more risks if we said that there's this special mechanism for developing or for running clinical trials in a harder to treat patient population, perhaps because the susceptibility thresholds that we think are there have already been exceeded. But if we fail in this, it will not have consequences on the main development program, and will not put the marketing of a drug into which hundreds of millions of dollars have been invested at risk, if that makes sense. And I don't know that I necessarily have an answer. I just pose the question: can we come up with a different way of perhaps doing clinical trials parallel to the main development program? Food for thought.
Food for thought. For later today or later meetings. But to then sum up this part of the meeting, I think we have some key takeaway lessons from our past experience. I think it came out very loud and clear that collaboration and data sharing and applying all the tools in terms of analytics and machine learning, et cetera, that we have will definitely be required. There's a lot we don't know that we need to start exploring, and that is different compartments and the evolution or the presence of variants that are neutralizable or not in these different compartments. Also, the other part that Joe raised is we don't quite understand yet, and the concern that you raised, Simon, the evolutionary, the drug pressure part of these antibodies, and to what extent they will then drive, the pre-existing variants to become the predominant, as well as through virus replication, generate new variants, obviously, that might become even less susceptible. So those are all questions we need to keep in mind. I think one big difference is that when we looked at all of these early resistance studies, we often had huge amounts of replicating viruses because patients weren't suppressed. So the generation of new mutations and new highly resistant viruses that, was really profound because there was a lot of replicating virus in the presence of drug pressure. And normally we wouldn't be doing the studies in that situation right now. So there's a lot we need to learn and remember and adapt, but then also really recognize the differences in the situation that we're in today with everything we've learned in the past. So thank you very much, Dan and Joe and Jackie, for your presentations, and the panel for all your comments. And everyone, I think we will I think we're right on time. We'll come back at 11. So enjoy your coffee and some snacks, and we'll see you back here. Thank you.
Thanks.
Test. All right, everyone, we want to be mindful of the time for our presenters, so we'll be getting started on session 2 in just 1 minute. And we will have an iPad for you once it's time for the panel discussion so that you're able to send a chat. Okay, you have to do that. Okay, yeah. Whenever you're ready.
Okay, everyone. Thanks for coming back from the break. My name is Boris Jürg from the Reagan Institute in Boston. And it's my pleasure now to moderate the second session. And we have a great lineup of speakers and panelists. We have 3 talks. And the topic is overview of bNAbs and development programs. So bNAbs that are currently really in advanced clinical development. And we have Peter Leone as the first speaker, who leads the clinical development of the N6LS program at ViiV. He's also professor of medicine. And we're very glad to have you, and I give you the stage.
So I'll be presenting this data on behalf of the N6LS team. It's not individual work at all. This, I know folks in the room know, but again, it's just to sort of orient you to where N6LS works. It acts at the CD4 super site. You can see the list of other bNAbs that also cover that same area. We've done our proof of concept, so I'll be able to talk about that. That was presented last year at Glasgow, and I'll double back over that. But we did show really good virologic response in an unscreened treatment-naive population. So for the proof of concept or the banner studies we'll talk about, that was a non-screened population. They were given a single dose of N6LS. And we saw a median decline in viremia of those individuals of 1.72 logs, and we saw a maximum of over 2.5 logs. So really good response to sensitive individuals. I'll touch on some of the sensitivity data that we have from both parts 1 and part 2 of Banner, and then some of the modeling data that we generated from that. So the Banner study was a randomized open label 2-part multi-center study. We wanted to look at safety, antiviral activity, and PK activity. Again, this is in treatment-naive individuals, no pre-screening. We looked at their baseline sensitivity afterwards. On the left-hand side, you can see the inclusion criteria. Based on feedback we got from the FDA, we actually did a virologically driven response in terms of the duration of monotherapy. So on the right, you can see what that monotherapy endpoint criteria was. So single dose, the dose ranging is listed in the middle there. So we went from 40 milligrams per kilogram down to approximately 1 milligram per kilogram IV. We also gave a subcutaneous administration of N6, which was at 700 milligrams. At that point, that was the largest single administration sub-Q of N6 without any additional enzymes to help. And I'll touch on that in a second, because I know Joe had brought up about volume, and there's some interesting information on that. The monotherapy endpoint criteria, again, given a dose, they were followed out to at least day 11. If they did not have a decline of a half log or more, that was considered a non-response, in which case they started on standard of care. We followed all those folks out for 48 weeks after starting standard of care. If they had more than a half log decline, we followed them out either to day 84, which no one hit, or if they had a return to within a half a log of their baseline viral load, or a rebound of a log or higher from their nadir. These were two treatment arms that we presented at Glasgow. We hope to present the remainder of this information from Banner at CROI this year. But I just wanted to point out a couple of things here. The 40 milligram per kilogram is the high dose. We actually gave a whole log lower dose at 4 milligrams to help to define, we thought, what that response would be. And to our surprise, we saw a good response. The bottom line is if you're sensitive to these bNAbs, you actually get a great virologic response. I'll spend a little bit of time talking about this one individual next. And I think it illustrates a couple of points. So this individual had over 2 log decline from their baseline, and they remained down. They weren't completely suppressed, but their drop, at least maximal drop, lasted till about day 70, and then they rebounded up. A couple of things to point out. Their viral load at baseline was relatively low at 5,000, and their sensitivity, this is an IC80 here, they were exquisitely sensitive. So there is something here to show that there are several factors that contribute to that. We presented this information last year at CROI looking at baseline sensitivity and viral load decline and time to rebound. And what you can see based on this, and it's not really obvious, if we look at the bottom, this is looking at going from very sensitive to least sensitive on the IC80 on the x-axis, and then the y-axis is going down. So the lower you are, the closer you are to the x-axis, the bigger the decline. And you can see a rough correlation here, moderate correlation of the more sensitive the virus, the more the decline. And the same thing in terms of time to rebound. This is looking at increasing days to rebound and sensitivity down here. The more sensitive, the longer it takes to rebound. So again, the phenotypic assay at least is helpful in telling us who we could treat and what kind of response we could expect. In terms of safety, we just presented this at EECS in Warsaw. From looking at Banner's part 1 and part 2, we saw no SAEs. We saw no drug-related grade 3 or grade 4 AEs. Of AEs that were grade 1 or 2 was only about 21%. All those were pretty mild. And I would point out that our subcutaneous administration here was really well tolerated, with only 4 grade 3 or grade 4 that, again, were not related to drug. And if we look at this subcu administration, there were actually 3 doses, subcutaneous administrations, in each one of these individuals. So there are 48 potential opportunities to see a reaction. We only saw in about 10% of those overall. If we look at taking the data and then looking at exposure and response, this was modeling that we presented at EECS in a poster as well. And you can see down at the bottom, looking at the concentration area under the curve here. So this is exposure to N6LS. And then looking at different IC90s, those are the lines that are shown here, and the different doses. The bottom line here, the take-home point, is that exposure is a pretty good predictor of response based on sensitivity. So as Joe had mentioned, and what I think you can take home from this is, you give more drug, you're going to see more coverage. The question is, what's a practical response of picking a dose? We don't have personalized medicine where each individual can be given a dose that's specific to them. And I think the challenge is, where's that clinical cutoff? Where do we draw the line based on the dose that we bring forward for development? So exposure is important. And if we look at different doses that were given IV, including this one, which was given subcut, you do get predictable exposures. This is in healthy volunteers. And if we look at the response here, you can see that the response, again, seems to be based on the exposure overall. So if you have a sensitive virus, if you give enough drug, you get a predictable response. All that is to say that that's led to now moving from our phase 2A study to a phase 2B trial that's now started. It's the EMBRACE study. We're recruiting only in the US. And the reason we're only in the US has to do with the IVDR issue in Europe. So I'm going to spike the ball and say that this is an important topic which we really need to get into. Our study is looking at a 2-drug combination of CAB plus N6 as a stable switch regimen. We will do pre-screening for these individuals, and to be eligible, their viral phenotypic sensitivity to N6, we're looking at an IC90 of less than 2 micrograms per mil or an MPI of greater than 98%. And we are using the monogram assay for that. It's broken up into 2 parts. In part 1, we have 2 different regimens of N6LS, IV that's given every 4 months, and then a subcutaneous administration here that, just to put things in perspective, it's 37.5 mils that we're giving, and we're giving it with pH20, which is a hyaluronidase enzyme that actually cleaves hyaline in a subcutaneous space. Doesn't create a pocket, but allows fluid to more rapidly enter into that space. We can give with pH20 that volume in about 12 minutes. Having seen people get this, it's incredibly well tolerated. And the only thing we've seen is erythema. You get a little bit of a puffy lump that then resolves within about 90 minutes, and you can't tell that there was anything administered. It's really pretty remarkable. After our primary endpoint, our hope is to actually match N6LS every 4 with a CAB formulation that can be matched to Q4. The plan is to move one of these into phase 3. We're probably not going to move both IV and subcut into a phase 3 study. So, single dose of N6 is well tolerated with few drug-related adverse events. They're mild, infusion, injection site reactions, no SAEs. In Banner Part 1, baseline viral sensitivity to N6LS correlated with magnitude and duration of antiviral response. Based on that, we're moving into our phase 2b trial, which has started. And I just want to bring up a couple of points that I hope we will continue to have in terms of discussion. One, which already came up, can we define more clinically relevant cutoffs for viral phenotypic sensitivity? Again, that 2 micrograms per mil or lower is an arbitrary cutoff that really came out of the ACTG study. Could it be 3 or 4 or 5? Don't know. I think so, but we don't know. We need to be able to move that forward somehow, but I think we're going to need help to define that in those populations. What is the relationship of viral sensitivity to vaccinal effect? I'm not sure they correlate. That's a broader question to be answered down the road. I know Ollie's on this call, so I'd be very curious to hear his take on that. Is a genotypic assay needed? And if so, is it even possible? The challenge for us is for N6LS, covers about 97–96% of isolates. That means only 2 or 3% are highly insensitive. Makes it really difficult to come up with enough samples to drive genotypic development. And I think that's a real barrier for us, is the broadness of the antibodies. And then finally, the IVDR impact on studies in the EU. And if the FDA were to adopt something like that, which I know there's been some discussion around that, could actually put a major block in moving this field forward. Again, I'm not looking for answers to these. I just wanted to put those out there for discussion. So that's it. I'm going to yield the floor to Boris, and I guess we'll have time for questions in the panel discussion.
Thank you so much. Questions? Ben. Yeah, you have to stay here. Yeah.
Just if you went back to your slide where you showed the relationship between susceptibility and change in virus load, I think that makes the point about how lack of diversity in the results can really impact our ability to do the analyses. Because as I recall the slide, you basically had a whole bunch of data clustered in the middle where you had not, this slide is very convincing. No, the previous one there, right? You have all these viruses that have within a log the same susceptibility, and yet you see between almost nothing and a 2.5 log decline. And if you didn't have those two outliers, there'd be no correlation whatsoever, right? So a different interpretation here would be, it's hard to say what the outcome is going to be given a certain susceptibility result. You really needed the exposure data, and that's where you had the very nice correlation.
Yeah, I do think it's a challenge. One, the N for part one is small. So we have what it is, you know, again, if you also look at the sensitivity distribution, there's clustering that occurs. It's really interesting. I don't know what it means. And that you've got this tight cluster and then you have these outliers. And again, I think future work to try to define those populations. But yes, I think this one causes one to pause a little bit. This helps a little bit more. And of course, we're looking at using this. This is based on viremic data with single drug therapy. The question is, and I know it came up earlier, what happens when you're using it in combination with something that has really a potent activity and a high genetic barrier? How much pull do you need with this second agent in this case being a bNAb in order to maintain suppression? And I don't know yet. That's why we're doing the studies.
So quite a few people have already discounted the option of different doses. And I think that's a shame because we've got examples where I think both Durinavir and dolutegravir were approved at treatment experienced and treatment naive doses, even though I think in the end they don't get used very much. But I wouldn't discount that option. If you found that there's broadly, you could define a sensitive person and a less sensitive person using two different doses. Shouldn't be necessarily.
Yeah, we've had a lot of internal discussions around that. So I tend to be in your camp. Ultimately, it's not my decision, but that maybe you would look at different dosing, especially since you're somewhat limited in this case with having to either give it IV or subcut. There would be that possibility to adjust dose, unlike, say, with a fixed pill.
So I know you hate that. All companies hate that complexity. But if you had two different IQs, or
yeah, I'm sure the commercial people, their hair's standing on end with my statement.
Joe Josh, communications for BVAT and living with HIV and other things. Are you looking at the reservoir at all? Are you looking at the impact? Because I think it's so
those
exciting.
are exploratory endpoints at this point. But yes, those studies are happening, and we'll see what we get with that. That was included in some of the stuff with Banner as well, some exploratory endpoints. And of course, the challenge is going to be following people out far enough to try to answer some of the questions. So that'll be forthcoming.
Yinda Huang again, HVTN. I'm a statistician, so I'm very curious. Like for this figure you're showing here, you have 4 different curves, each one for a different values of IC80, different susceptibility. If you divide the x-axis by the IC80 over there, you might get a uniform curve for all the scenarios. And that's kind of what I like to talk about in the afternoon. That's a variant of a predicted in vivo neutralization titer.
No, I think it'd be great. So the more input, because I am definitely not a statistician. So I think that's the kind of things that we need to be able to have and look at. Yes.
Thanks. One last question for you, actually from my side. So the rebound viruses, how did they look like in terms of susceptibility or escape mutations that you discovered or saw?
Well, I know Margaret is on the call. So Margaret Gartland has been looking at that. We are looking at it, and you do see a shift. What I can tell you is you see a change in sensitivity. What that means and whether or not that's clinically relevant down the road and whether it persists is something that we still have to answer. But what's interesting to me is there's something different between those bNAbs that act on the CD4 binding site with rebound and what you see compared to some of the other sites on the envelope. And I don't understand exactly what that difference is, but we did see a shift.
Great, thank you very much. So let's move on to the next speaker, which is Marina Kasky. And I guess she's going to present online, as far as I understand. She's a professor of clinical investigation at Rockefeller, and we all know she's done a lot of work with 3BNC 117 and 1074.
Hello. In the afternoon. Let's see if I can, oh, now I can share my screen. Okay, so my disclaimer is that I not developing the antibodies as commercial products. This is Sean's job. So what I'm going to be doing here today is to talk about 3BNC 117LS and then really both 3BNC and 1074, the work that we have done, because the two antibodies are being developed in combination. So just going back to old data now. So as we all know, each of these antibodies have been initially characterized by their in vitro neutralizing activity against different pseudovirus panels. And in the case of 3BNC and 1074, we are able to achieve high coverage with the combination of the two antibodies, where in this particular panel, there was coverage of over 90% with either antibody, but only about 43%, I believe, with the two antibodies. And then in this case, sensitivity was called as less than 10 micrograms per ml. Subsequently, because when we started to have our experience clinically, we started to wonder how the pseudovirus panels compared if we were to test viruses that were emerging from PBMCs. And this is a study that Julio, when he was in Michelle's lab here, he tested primary isolates from clades A, C, and D against this panel of antibodies. And then what he found typically is that the neutralization sensitivity of primary isolates were in general lower than pseudoviruses. But there was no correction factor that could be applied across the board for different clades or different viruses even. So we noticed that there are these differences between pseudoviruses and primary isolates. And I think, as we have also talked about already here today, is that while in vitro neutralization data allows the selection of promising candidates, we have all been learning that in vivo responses are way more complex and then really relate to the diversity of sensitivity of viruses that can emerge from the reservoir or they're in circulation and exposure to the antibodies. So these two antibodies have been tested in a series of small phase 1 type studies so far. They have been given both sub-Q and with IV administration. Sub-Q, we have not used the enzyme that allows for higher doses, so we don't have experience with that yet. And then they've shown antiviral activity in different settings. So I just summarize here in the bottom that when they were given in the context of viremic individuals, we saw also transient decline in viremia that was followed by going back to baseline in the majority of the participants with selection of resistance more clearly to 1074 than to 3BNC. In the context of ART suppression in switch studies, we found that if the participant had sensitive viruses to begin with, continued exposure to the antibodies was able to maintain suppression. The antibodies have been modified to have the LS mutations, and they have good half-lives, with 3BNC showing a half-life of about 66 days and 1074 or 80 days. So these are studies that were published last year. Here on the left is the study that we did here at Rockefeller, and then Tae-Wook Chun did another one at NIH. The participants were all virologically suppressed on ART, and they transitioned to bNAb infusions over the course of 20 or 24 weeks. And what we found, these participants were not screened for sensitivity to the antibodies, is that for both studies, we found more or less 70% suppression during the dosing period. And this is a summary of the sensitivity data that we then did post-hoc using the PhenoSense assay, but also genotypically here in the lab. And then Sean alluded to this earlier today. So I know that this is very busy. The bars here are the time to viral rebound for these 16 participants. Here in the middle is the PhenoSense assay results, where I put the actual IC90s, and then red and green here mean that it's defined as resistant if they had an IC90 more than 1 microgram per ml, or green less than 1 microgram per ml. And gray were the times when we were not able to have a result. So first, that's one point to make, is that we are all learning that sometimes the PhenoSense assay doesn't yield a result, and that can be in the range of about 20%. But we also had trouble with some of the sequencing looking for 1074 mutations. And then the other point to make is that while for some participants that rebounded early, we were able to detect resistance by PhenoSense, there was a mismatch of what we could predict in terms of 1074 by sequencing. There were several examples where the participants maintained suppression for over 24, 30, or longer weeks, and PhenoSense would have predicted that they had resistance to both or to one or the other antibody. So again, we still don't understand exactly how to use the assay clinically, or what the threshold should be, how predictive they really are. We are now having another study here where we are enrolling participants without screening, and we are seeing similar patterns once again. So that's the data in terms of viremia, maintaining suppression. But across the various studies that have been done with 3BNC and 1074, we also had some cases where we think that there was post-treatment control in some of the participants that received antibodies. But the studies are all very small. I'm highlighting here a study by Ole's group in Denmark, eClear. And this is the time to viral rebound. So in this study, the participants were ART naive, and they received 3BNC plus minus romidepsin at ART start, and they discontinued ART 1 year later when the antibody was cleared. And then what the curve is showing here is that the participants that had resistance, so neutralization resistance by PhenoSense in the plasma viruses prior to ART, they had viral rebound in the typical manner where it emerges in a few weeks after ATI, and it's fast in its growth curve. Whereas the participants that had sensitivity to the antibody, as defined in the study, they had one maintained suppression, the other ones had this different viral dynamics of return of viremia. But the reality is all of these studies have been very small. We don't really understand if these responses had anything really directly to do with the antibodies, if there was a vaccinal effect. And that's why we are now focusing on reproducing some of those results in other studies. And in addition to that, we know that the results haven't been perfect. We can improve on them. And then we are testing different combinations in different clinical scenarios. So to summarize across the board in prevention, these antibodies could be prevention and therapy. They could be perhaps alternatives to long-acting ARV, and there may be some clinical scenarios where they may be of greater interest. We are not pursuing a prevention efficacy study at this point, but there is this study that I'll talk in the next slide where we are trying to use this type of study to learn information that we believe can be useful to the prevention field as well. And then, as Joe alluded to earlier, Gilead and Sean's program, they're pursuing the use of the two antibodies in combination with their capsid inhibitor in switch studies, possibly as a long-term, long-acting therapy. And then in here, we are testing different combinations of antibodies with vaccines and other immunomodulatory molecules, because we are trying to engage what we know from cancer immunotherapy that antibodies can do, which is to engage through Fc effector functions and then perhaps have an effect in HIV persistence. So when we are thinking about using the antibodies to guide prevention studies, what I mean by that? We think that there are parallels that one can draw from what we learn in an ATI study by studying the rebound viruses in relationship to the antibodies that are on board at that given time. And this has parallels to the moment of HIV acquisition at a certain antibody level and the exposing viruses that break through. So this is the study that the ACTG is doing with the VTN and the PTN. It's a simple ATI study where participants are going to receive the two antibodies or placebo. They're going to discontinue ART, and they're going to be followed through viral rebound. Here, the study is going to happen in the Clade C area of the world. So this is the first time the antibodies are going to be taken to Sub-Saharan Africa. in this context. And then in addition to looking at virus suppression, as I mentioned, what we are really interested is to see if we can learn what's the minimum required serum concentration to maintain virus suppression. More broadly, the antibodies, when we are thinking about their use for acute or long-term virologic control, there are a series of studies in primary or early HIV infection, where the antibodies are going to be given at ART initiation. The RIO is an ongoing study that is being led by Sarah Fiedler, where the antibodies are given in individuals that started primary infection during ART suppression or in the placebo group when they reinitiate ART. And also the ACTG just opened A5374 that tests, again, in a population that started ART during primary infection, the two antibodies in combination of two vaccines and a TLR7 agonist. In addition, we have another study in sub-Saharan Africa where the antibodies are going to be given in a population of ART naive individuals. And then we have studies in chronic infection where we are trying to reproduce the findings that we had that maybe the antibodies had an effect in the viral reservoir by prolonging the exposure to the antibodies during suppressive ART. And then another study where the antibodies are being given with an IL-15 superagonist in the context of ATI. And Sarah and John are now going to be working on a study of the two antibodies with a combination of two other vaccines. So to summarize, with these two studies, we have seen that we had transient decline in viremic studies. We were able to maintain virus suppression and participants had sensitive viruses. The challenge that we've been hearing all along today is identifying who those participants are. We have seen effects in terms of immune responses and also in the reservoir, but these have been small effects that have to be studied further in larger studies. And these are all of our collaborators, and we couldn't do any of this without study participants. Thank you.
Thank you, Marina. Did you hear the applause? She didn't. Can you hear me?
Yes.
Okay. Thanks so much. Are there any questions here from you folks? Dan has a question.
I'll just make a quick comment. I think that as you pointed out, Marina, all these studies are very small, and you can see different associations between susceptibility testing and outcome depending on which cohorts you're looking at. In the study that Matthias, Roger, and I did with the infants, it looked like there was a correlation that the viruses or proviruses tested retrospectively that showed susceptibility predicted babies who would remain suppressed on the combination of, in that case, 10-1074 and VRC01, not the best antibody to use in subtype C. And those that were resistant almost uniformly failed. But again, that was a small group of about 25 kids, 40% of whom maintained suppression. So I think we need much larger data sets to be able to say what value the predictive is, what the predictive value of these tests is going to be with particular antibodies in specific populations.
Yeah, and I completely agree. And I also think then that in that population, it may be that the population like the infants that were treated very early, so they had what we think less diversity in the reservoir, the assays would be more likely to be predictive.
Absolutely. I think that's a key point. Yes, thank you for making that. Yes, they most certainly had much less viral diversity in their population.
Okay, I don't see any other comments. Thanks, Marina, and we're going to talk in a bit on the panel. So our third speaker, and I hope I don't butcher the last name, but it's Rip Ballou, okay, who is the project lead for HIV bNAbs at IAVI, long history at GSK, a global head for vaccines there, and you worked also at the Gates Foundation. Great, thanks for having me.
Thank you very much for inviting me. I'm a little bit at a disadvantage here because I am actually not an HIV expert. My background is primarily in vaccines, and I spent a lot of career working on malaria vaccines. But I joined IAVI a few years ago and have been working to move some very interesting bNAbs into the clinic. And I don't have any clinical data to show you because we are still in the manufacturing stage of our antibodies. But I think that the work that we're doing is potentially of big interest to the treatment and cure community. We are a nonprofit organization and depend upon the goodwill of our donors to support the work that we do. Our mission is to translate scientific discoveries into affordable, globally accessible public health solutions, and this is particularly challenging in the area of monoclonal antibodies. The work that we have done has been to try and move the field of monoclonal antibody development so that this issue of accessibility is addressed early on. And it's been primarily by working on increasing the potency of these antibodies. Our programs at IAVI fall into two big categories: vaccine work shown on the right and monoclonal antibody work on the left. The HIV monoclonal antibody work is by far the most advanced on the bNAb side. And on the HIV vaccine side, we've been working, of course, with the germline targeting approach, which is the whole concept has come out of the identification of targets that bNAbs recognize. And the germline targeting vaccination strategy is a way to actively elicit those bNAb responses in humans. And we have a late-stage program in Lassa fever and in tuberculosis. One of the things that we have learned over the years, and these are data coming from non-human primates, is that there is a correlation between serum titers required for complete protection against HIV challenge and their potency. And the strategy that IAVI has used, and this differs from the way the VRC has used to develop their VRC01 class antibodies into much more potent antibodies. At IAVI, we've been using essentially yeast display and directed evolution. The idea being you take the parental antibody, create a mutagenesis library, select for high affinity variants, characterize these for mutations that enhance affinity, and then down select based upon neutralization and developability criteria and this has been done for the PGT121 parental and the PGDM 1400. I would point out that both of those antibodies came from a USAID funded protocol G that was started more than 20 years ago. In fact, 1074 came out of that same population of volunteers. And these, we have worked on those. Those antibodies have been obviously made available to a number of investigators, talking about the parentals, and they have gone into the clinic and into the pediatric space in both combinations and individually. We have focused at IAVI on trying to take that 121 and the 1400 and make them significantly more potent. And if you look now at this mock-up here, this gives you a sense of the difference in potency and breadth that we see with these engineered antibodies. Certainly the CD4 binding site class antibodies, the VRC01 class, we saw several of them earlier. Exceptional breadth, but not particularly potent. And for IV administration, it's probably not a big deal. You can give lots of antibody IV. For our target populations in sub-Saharan Africa, IV administration may be technically feasible, but the cost of goods is going to be prohibitive if we have to give grams and grams of antibodies. And so we're very concerned about potency. The V3 glycan antibody, the engineered 121, 50 to 60% breadth, but potency in the 1 to 10 nanogram per mil level, similarly for the 1400. And these are the potencies, the levels of potency that the licensed SARS-CoV-2 antibodies that were given predominantly sub-Q or IM have been able to achieve and became commercially viable products that did not require intravenous administration. So when we look at the work, and this work has all been done by Devin Sock. I'm really here speaking on his behalf at the Scripps. He's an Ivy scientist who works at our laboratory at the Scripps and discovered these antibodies and has now engineered them to what we call second generation antibodies. And the combination that we are looking to take into the clinic is a combination of a engineered 121, PGDM 1400, and a more recently developed antibody from the VRC called VRC01.23LS. Just to give you a sense of the increased potency that we can see, this is now against clade C viruses isolated from the AMP trial. When these three antibodies are used together, more than 95% of them are neutralized at a very stringent cutoff of 0.3 micrograms per mil. And if you look at B viruses from the AMP trial, it's 88%. And you contrast that with the activity that we see in the parentals, the 121, for example, 33% here. And the engineered going up to 69%. So quite a bit increase in potency and breadth by this engineering approach. And interestingly, the EPGT-121 seems to have a greater level of ADCC activity compared to the parental antibodies. And this may, again, play to this, quote, vaccinal effect that we might be seeing with these antibodies in a treatment setting. So where we are right now with our program, VRC01.23LS has been produced by the VRC under contract and is in a phase 1 clinical trial right now. We have evidence of good safety. We don't have PK data from that study. These are all LS modified antibodies, so we're hoping for half-lives in the 60-80 day range. The 121 GMP production is well underway. We hope to be at the final stages of purification and filling our vials in the coming weeks. And we're expecting that clinical trial material to be available for a phase 1 study that could start in combination with VRC01 in early Q2 of next year. Now, one of the issues that has come up during the development of these engineered antibodies is that it has been difficult to predict from the early development cycles what the actual manufacturability of these antibodies has. And we have run into a challenge with the EPGDM 1400 V9. This cell line, during cell line development, looked as if it was going to be developable in terms of its yield. But when we put this through extensive engineering development work at our CDMO, really could never get the yield of this antibody much above 3 grams per liter, which frankly is not a commercially viable level of production. And so we regrettably had to abandon that particular cell line. We have looked at a number of parameters around this in both perfusion and batch fed production methodologies. It doesn't seem to be a media-driven issue, so there may be some inherent toxicity of this antibody to the specific cell that was selected for cell line development, the clone that was selected for cell line development. But we don't have direct data of that point. We are moving to another version of PGDM 1400. It's called .93LS, developed by Dan Baruch and colleagues at Just Bio, which was essentially the parental PGDM that has been modified to enhance its productivity and manufacturability. And from what we hear, this looks to be producing in the range of 8 to 10 grams per liter, at least at a small scale. So the NIH is working on the development of that particular clinical trial material. We have a concept protocol that has been approved, and we will do full protocol development once we have all the certificates analysis on the 121. And the intent would be to start with 2 antibodies and then bring in the 1400 when the clinical trial materials available. And, you know, given the potency and the breadth of these antibodies when used together, we do think that these engineered antibodies, particularly the 121, could play a role in treatment and cure indications. So I'll stop there. Thank you.
Great, thank you very much. Any questions in the room right now?
Hi, Kalyan Pandey from Merck Biologics Discovery. A really nice talk. So you talked about the potency enhancement with some of these antibodies. So how much of the biophysical characterization, developability work that you guys had to do to isolate your final candidates?
I'm not sure I understood the question. Are you talking about the engineering work that had to do at our CDMO?
Yeah, often when you are enhancing the affinity of this antibody, it comes up with the liabilities.
Yes.
With the, the developability properties of those antibodies. So, how much of that work did you guys have to do?
I mean, it's interesting. The way that we did this work was in collaboration with the VRC. And we had the initial AMBER screening of essentially cell lines done to down select to cell lines that we thought could be developable. We actually had another version of the BRC01.23LS that had a just bio modification in it that looked quite good. But when we got to the point of post purification, there was this unexpected phase separation that we could not resolve, and we had to abandon that. So that was, a year's worth of work and then pick up an alternative. In the case of the PGDM 1400 engineered version, there was almost 2 years of engineering work done on that to try and sort that out, and work that we're doing with another CDMO on perfusion technology doesn't seem to have solved it. So these are very complex developmental challenges. They are important for us because we really have a volume issue and a cost of goods issue with our access commitments on this. And so spending the time up front to get these right is really important.
Thank you.
Yeah, hi, Joe, you're on. Thanks. That was really a remarkable talk. It went by kind of fast. Were the PGT 121 and the PGDM 1400 specifically modified towards subtype C? Is that in the kind of enhancement process or whatever you call it, the improving affinity? Was that specifically targeting subtype?
These are randomly introduced mutations,
right,
and then screening against the virus isolates that were C and B, because they came out of the AMP study.
Oh, C and B. Okay.
These are isolates that came out of the AMP study.
Because it looked like the combined activity was actually slightly better against C than B. Did I see that correctly?
And these samples, the original isolates came from subjects in Protocol G, which were from Sub-Saharan Africa.
Sub-Saharan Africa. Great. That's really helpful. I think this particular combination is really fascinating for, where one could imagine using this combination, if we're talking about the US, which you're not, but I guess we are, to actually treat without screening, and maybe even people who are viremic, or thinking about a study that would try to investigate that question, on a very, very small scale, but with really, careful guardrails. I think that would be, and these you think are going to be every 6 monthly? Is that your guess?
I think it really depends upon the PK. I mean,
you don't really know yet. Sorry, my mistake. Right.
Is not having. I mean, we do think that you need monotherapy isn't going to work in the setting for prophylaxis.
Sure.
And we are focusing on the infant target population, which maybe has some, as we've heard, less
Right.
complex virus beginning. But it's important to not have, a single antibody uncovered. And so when we look at the individual PKs and then giving them together, it would be great if we could get to 3 to 4 month dosing. I think it really depends upon
what I forgot
what trough we can get given the volume restrictions that we can give a child.
And then one last question, I'm sorry. Like when you do this maturation and increase affinity, does that change your risk of anti-drug antibody, or it's really, who knows?
The interesting thing is that this 1400 is extensively modified, and we did take it through a full tox profile, and there was no ADA even in rats. We did see ADA in rats with 121, but 121, that's not unexpected.
Sure. Wow.
We'll see what we get in the clinic.
There's one question in the chat.
Hi, thanks everyone. Hoping you can
We can hear you.
me. Okay, wonderful. Thank you. So thanks for acknowledging the problem with the cost of goods. IAVI has been very helpful in shedding light on this, as well as animating conversations with other developers on what could be done. And I want to flag why it matters upstream. You said obviously you're looking at higher dose, you're looking at longer half-life. And part of it is that already R&D for and using antibodies is driven by the cost. So there are some products, for example, malaria, and IAVI, I would love for you to sound in on this, where the use cases are being drafted partially out of need, absolutely, but also because of costs. The rationale is because the cost of goods is so high, maybe at least we can get it going for lower doses, and that's children who require lower doses. So it would be great to hear from you whether that also plays out in terms of the use case here. So the malaria is also for prevention. It's also safe for women, pregnant women and children under 5. And those two populations are at high risk during malaria seasons.
You're talking about the malaria monoclonal, correct?
Yeah, but part of the rollout and the use case that is being drafted and where we might find, real implementation is limited to children. I hope it won't stay that way, but again, it's because the cost. So thank you again for raising that.
Yeah, the dosing, the data from the clinical trial that showed efficacy in West Africa with the malaria monoclonal showed a difference between 10 mg per kilogram and 40 mg per kilogram. And, I think that antibody, it's the same target as the vaccines that have currently been licensed, and the R21 will be licensed almost certainly. And to me, the issue with that antibody is it's not potent enough, and it needs the engineering that we've done on the HIV side. Shelley Malhotra from our access group at IAVI has really been leading this discussion, and the use case for HIV in the pediatric space is really quite impressive in terms of its cost effectiveness. So I think that, and it's driven by the fact that we hope to be able to use small doses at reasonable intervals that could be given like vaccines.
Thank you for that. Just on the malaria, it is a 6-month protection, 1 shot subcutaneous. So the administration is actually less than the current vaccine. Over.
Great, thank you very much. So I would like to invite the speakers to come for the panel discussion over here. And then we have two more panelists, Richard Jeffries from the Treatment Action Group, who runs or leads the Basic Science Vaccines and Cure projects, and Damon Deming from the FDA. Okay, wonderful. Everyone is here. So very interesting. A lot of stuff happening. You know, I mean, Marina has this slide where she has these groups of prevention, therapy, cure. So I think we're not completely set yet where they fit the best. Maybe they fit everywhere, maybe not. Obviously, there's always the discussion. An infusion monoclonal antibody is definitely more expensive than an antiretroviral therapy, and we know already that works fantastic. And we have the long-acting versions of those. So you know, the question is always in what niche do these antibodies fit? And I would like to just get some impressions from you guys on that. I don't know, Peter, do you want to start maybe?
I mean, we're moving forward, obviously, in treatment suppressed individuals, but I do think the excitement is in the stuff that you talked about and what Dan was talking about in the population with neonates, both in terms of prevention and treatment, given the safety profile and the potential half-life for these things really opens up a world of possibilities. It's just that we have to do that in collaboration. So to me, there should be a full court press to work on that simultaneously. We shouldn't be sequenced off, but that to me is the most exciting part of it.
As I said, IAVI has really looked at the pediatric peri and postnatal transmission targets as being a sort of a sweet spot where there's an unmet medical need. There's 130,000 kids per year who are becoming infected, and that's in the context of ART programs. And there's many reasons why that occurs, not the least of which is that a number of those infections occur after delivery during breastfeeding, and from mothers who were not infected at the time of the delivery, but become infected during the 18, 24 months of breastfeeding. And so we do think this is a specific area that isn't going to go away. The development of the long-acting antiretrovirals for the pediatric population is progressing, but it's like many other drugs, it's progressing after all the adult indications.
Yeah, and just to add on to that, I mean, the idea that you could give a baby at birth a single shot that could protect them for 6-9 months to sort of bridge that gap is really, it's a game changer. And I mean, it wouldn't prevent us from doing all the other things in terms of treatment of the mother and prevention, but it's that gap. It's just, we need resources and help to be able to do that.
Richard, do you want to comment on the community aspect and how this is actually accepted or seen right now as a potential treatment or add-on replacement for ARVs?
Sure thing. Obviously, I can't speak broadly to the community. I think it would be important to do some social science to help better understand what the sort of priorities that people have, particularly people participating in the studies, how they're reacting to the kinds of administration, dosing, and so on. You know, one area that has been brought up, there was a study from the Institute for Human Virology, I think, suggesting that maybe bNAbs would be a good option for people if they need treatment for cancer, that there would be less interactions with antiretrovirals. So I know there's some interest in those kind of uses. I just read, I don't know if you know Tim Murphy, who writes for The Body, just wrote a story about how he required emergency surgery and his HIV treatments got mixed up and he ended up getting resistant to all of the components of his regimen. So I don't know if bNAbs would be an option for people undergoing surgery that that might need a different regimen from ARVs to be less of a concern with interactions and so on.
I think it's interesting to hear. I've been sort of shocked at what I've looked at in terms of the issues that the RSV monoclonal nirsevimab, which was actually developed by my new IAVI colleague, John Heinrichs, is here in the audience. That on the internet, when they talk about the shortage of that monoclonal antibody, they talk about the vaccine shortage. It's being discussed in the United States as a vaccine, not as a monoclonal antibody. And that, at least in this pediatric setting, the idea that it would be given as a injection of a mil or less on a schedule that looks like a vaccine schedule, I think from a community perspective, the idea of talking about it as a vaccine, as opposed to something else that they have to understand and have to worry about, is particularly if it has a safety profile that we think that they should. I think this is an untapped area that we should be thinking about.
Yeah, language matters.
I was going to push back on that a little bit, just because I think you want people to understand the reality of what's happening, and there is a difference between active and passive immunization, and I think it's important people do understand that, even though it's serving such a similar purpose.
Yeah, no, I mean, I fully agree with that. But there's been so much, especially in the US, resistance to, when we start talking about some of these things, how we phrase it or how we communicate, that's important. But no one's advocating we don't provide full information.
It's difficult to adapt to irrationality, I think. The one other thing I did just want to mention, I think people are always interested in knowing who's participating in the studies. So a quick demographics slide, because we talked about a number of studies in these presentations without actually talking about who was in them.
Yeah, for the sake of time, I cut my demographics, so I'm happy to send that to you.
I mean, I think like monoclonals against infectious diseases obviously became pretty popular, or at least known to the greater population of COVID now, because before, first of all, we didn't really have much RSV maybe, but there was not much else around. And then COVID brought in these antibodies. They were hyped up. People got them, but they also read pretty quickly that, yeah, escape, couldn't use it anymore. So, the question is a little bit like, what's the feeling right now in the population on monoclonals against an infection? And, maybe they're more familiar with the concept overall now, potentially, but also the bad press that it got with the rapid escape. Are people comfortable doing that now for HIV, where they know that escape is really nothing they want to have? I mean, I don't know. I cannot speak. I don't know for the population or for the people, but I don't know what you guys' impression is on that.
Yeah, can I jump in here?
Yeah, sure.
So John Heinrichs, IAVI, just a few comments on the development of nirsevimab. I think it was, from our point of view, when I was at Sanofi, very important to try and develop nirsevimab in a way that was akin to a vaccine, because we wanted to gain access to the entire infant population. And at least in the US, to do that, it was critical to get that listed on the VFC, the Vaccines for Children program, so that we would get reimbursement for all infants. So that's one comment I'd make about why that was the tact that we used for development of that antibody. In terms of protection, maybe one lesson we can learn from nirsevimab was that we evaluated protection over a 150-day period, so 5-month period, which was coincidental with the RSV season, and we demonstrated that protection across that window. But when we actually looked at neutralizing antibody titers in that antibody in treated children, we actually were able to maintain antibody levels that were 9-fold higher than baseline at day 360, so for a full year. And they were even higher at that time point than convalescent sera in infants that were in the placebo group that had become infected with RSV. So I think that there's an opportunity here with some of these antibodies to go even well beyond what we consider as that typical protection window. Thanks.
Great, thank you. So just from the chat, Sarah Fiddler says that actually using bNAbs to avoid complex drug-drug interactions is a relevant point. I just want to move on quickly to, I mean, you guys developing the antibodies further. We tested the parental versions. Now the, the modified versions, they're becoming broader. So breadth obviously is always kind of the goal, given that we have that viral diversity and the rapid escape and selection and all that. Half-life is the other thing that people work on. We can come back to FC modifications a little bit later, maybe, but question maybe for what the FDA will think, and that's for Damon. What is kind of the cutoff of breadth that we are looking for? What is the need kind of, or how we define that? I know it's a difficult question, I don't think there is the answer for, but what is expected?
There is no expectation. I mean, it's going to depend on what the final product looks like, what's the combination can cover. And keep in mind, these estimates for coverage, we don't even know how good they actually are real world. Or even the activity. So it's one thing to compare antibodies with the neutralization assay, but as everyone knows, they're multifunctional molecules. So when you put those other potential pathways for activity in vivo, that rank might change. So it would just, we try to keep this all in mind as we review the data. So right now we're right there with you. We're just learning and following with interest. It's very interesting.
So the question is the down selection process, because we have to down select antibodies. We cannot test every variant in humans at large scale. So we have to come up with something. It's IC50, IC90. Should we add another level of FC function or something on top and make it a little bit more holistic kind of before we move forward? Or
that you would quickly approach the point where it's not practical. I think that the approach so far has been reasonable, just looking for the most potent neutralizing, but just keep in mind that these cross-molecule comparisons might not be as easy as we might hope. So in terms of, there's little reason to think that FC effector function is going to reduce the relative in vivo activity beyond what you're just getting with neutralization. So it's not a bad thing to ignore, at least early on, but it is something to think of. That was a terrible answer. So basically, there is nothing prescribed, and we're not sure. We're just following it with interest.
So it gives everyone a lot of freedom.
Yes, absolutely. This is still very much in the early exploratory phase, I would say.
Okay. Coming back to the FC, because you mentioned it, and Marina also mentioned it, we don't even know completely, if you think about therapy or cure or eradication of the viral reservoir, we don't really know exactly what the mechanisms are. Are the bNAbs the right antibodies for that? Do we need something else? And it's being tested in vitro, but overall not really advanced. I mean, FC modifications for cancer antibodies is something that is more common and more standard and being pursued. For cure, is that something that should be specifically developed, or I don't know who wants to take that one?
I think it comes back to we don't know. I think we talk about cure, it's a complicated space. It's going to require multiple approaches. And we still tend to think in boxes in terms of treatment versus cure. So I suspect that's going to be different, a different need, probably an and rather than an or.
I think it'd be kind of interesting to look at, and it sounds like you already are, whether there's any kind of trimming of the reservoir happening in the treatment context. In case that might have sort of inflammatory or anti-inflammatory activity or something like that.
Maybe this one.
They just wanted people to exercise.
Yeah, no, I get my steps in. Wait, set my watch. I mean, I think pragmatically, Boris, it just seems to me that companies are going to develop stuff for treatment, I think. And if they happen to have, a cure side effect. I think that's the more likely, at least over the short term, right? If we get closer and actually something looks like it could be modified and actually affect a true or complete or functional cure, then that seems more likely. The thing, Peter, I hope you guys are doing is this issue, which I'm sure Boris could talk more about, is this idea that somehow these antibodies actually change CD4 or CD8 activity, right? I mean, that's at least in animals that's been seen, right? For sure, right? In animals, and especially in the people who received it and were viremic, because again, that's I think the SHIV model, right, is where they actually saw induction of specific CD8 immunity that could be knocked down, and then so those people would be, I think, those participants who volunteered for the study. Their samples might be particularly valuable in terms of knowing whether there was some impact on not only on the reservoir, which I think is somewhat less likely, but actually on the immune response, and so that would be something worth exploring, I think. And but Boris could probably talk more about that than me.
Yeah, I mean, there's obviously quite some data on that. I mean, Michelle Nussenzweig's group had shown a boost of autologous antibody responses after bNAb therapy, and then Daniel Kaufman and Marina can talk more about it because she's still here. Showed a boost of T cell immunity pretty much. I think the idea is always that you have some virus bound to your antibodies, these immune complexes are being presented, and therefore expand kind of T cell or also autologous antibody response. And that this kind of vaccinal effect might contribute also to some form of viral control. At least in the monkeys, it was pretty clear. If you knocked out the CD8s, the monkeys lost control pretty much. Marina, do you want to comment on that aspect of the bNAbs?
Yeah, I think, and to add to those studies, that is also the data from Ola's group with eClear that also showed it seemed to be that there was a relationship with the CD8 T cell responses in those interesting delayed virologic rebound curves. So I think that if we look at it collectively, there are these interesting participants in all of these small studies so far. I think it'll be really important to understand what about those CD8 those T cell responses may really be involved in control. Not only, I think, characterizing their functionality, but really, I think also the specificity. What about where in the virus is it that they are recognized? And, is the antibody really playing a role in how these shifts are happening or not happening? Which goes back to what Joe said, that for all of these studies, I think it's really important to have the samples to go back to these very interesting participants. Not only the studies that we do in academia, but you guys developing, if you keep that in mind, that would be really extremely helpful to all of us.
Thank you, Marina. I see that Shannon has raised her hand. Can you hear us? Okay. I think there was a comment that you wanted to show a video from Tony Fauci about
Sorry,
antibodies.
I didn't realize you were calling on me. I have
sorry.
no, it's okay. I just have 3 comments. One is in terms of the community response. I have to say that part of the community response to the hope of any therapeutic or prevention MAB or bNAbs will be met with expectation and relief that the HIV largesse is going to be brought to bear to make MABs effective, suitable, affordable, available. That infectious disease attention for HIV, for malaria, for RSV, those important MABs or antibodies that are in the pipeline can lift all boats. that's a hope that I hope isn't misplaced, because what the community will want is also access to the NCD MAbs. You know, there's trastuzumab that was approved by US FDA in 1998. Oncologists, some referred to it as, you know, revolutionary for breast cancer. But it's barely anywhere, even though, again, the approval was in 1998. I also just want to caution against, as we go further, there are some developers that already draw the line on not furthering investigations or development when the delivery is intravenous infusions. I don't want us to make the same mistake as we did in 1998 and 99 when we started talking about ARVs for the global south. We can't pretend that all of Africa is a desert. So in this instance, of course, let's go for suitability, but also let's not let the perfect be the enemy of the good. And finally, yes, I will share the video from Fauci. It was for a meeting where we convened 8 US government agencies to talk about their work on antibodies and to share the optimization agenda, because each one was looking at how to make what hasn't been possible. So I will share that in the chat. Thank you.
Thank you.
And I think maybe that, you know, it highlights, I didn't necessarily answer the question, your first question all that well, because I think, you know, clearly there is community interest and it's driving community advocacy already. And some of that I think that Sharon Ann is involved in, around how we prepare for the possibility that these become useful and how to ensure that they don't become another sort of member of the sort of two-tier therapeutic landscape that we have for so many other things like CAR T cells and so on.
Sam. Thanks. So I always support and defer to Sharon Ann's points about access, because they are essential. Really. You know, we're at a time now where TLD, which is first line best combination, is now available at less than $50 a year, you know, which was unthinkable, certainly for most of the treatment time we've been treating HIV. I've got, like, almost of my questions are all simple questions, but I did go to the NIH, do excellent bNAb meetings up at the campus up in Maryland a few years ago. And one of the important points there, or optimistic points there, was that there will always be new antibodies and there will always be better antibodies, and that the field will continually discover, it's taken for granted, better and new antibodies. But it also depends on where you're looking for them, really. And it sounded like IAVI, you're looking at, you're going to source antibodies from different clades other than subtype B. And whether we're missing something, whether we might find monoclonal antibodies that are coming from people who have very strong immune responses to subtype C, and that they might be more potent against subtype B. So that's just like a general question.
Well, I mean, I think that point is very well made. The IAVI has actually made the effort to move the technology for engineering these antibodies to our laboratory in India, where they have very different distribution of clades. And this parallel work to look for very potent monoclonals that could be further engineered from those populations is ongoing. It's lagged behind the work that has been done to date with the samples that have largely come for IAVI from Protocol G, which I said these samples were collected 20 years ago.
But Protocol G was primarily African continent?
Yes. Yeah.
And I think maybe shout out for the study participants from whom these antibodies came, who may not realize that they're now being developed by bNAbs, for example.
Lucio?
I'm Lucio Gama from the Vaccine Research Center. I'm a little bit concerned about like 3 points that I see that at times they don't get along very well. First is that we do not understand very well where those antibodies are going. how long they're going to stay in tissue. I know that in HVTN, it just did like 2 beautiful studies, but we still don't know when each antibody is going to work and how they're going to work. Each one has their own personality, and Rip knows about that. You know, I mean, you make one, you think it's going to be good, and then you fail, and then you put in people and have adverse effects. So we still have a lot of things to learn about antibodies, and we keep making more antibodies all the time. So where's going to be like this balance where we can really push from one moment where we say, well, now we have the antibodies. Now we have 3 antibodies that we can use, and we can at least like do a lot of studies with those antibodies. We have already, like, you know, VRC07 for 23LS, PGT121 for 14LS, and PGTM for 300LS. They're being tested in a lot of situations. Is that enough, or we're going to need more and more and more? How much is going to be enough so that perfect does not interfere with very good?
Don't know the answer to that. I mean, I think you try to move the field forward. I still view what we're doing, even though ultimately we want to launch a product, is to contribute to the field as it moves forward. But, you know, HIV is the dynamic infection. And I think, I know Joe and I were talking about this, sort of the coverage that you get maybe shifting in the population based on what's being transmitted. So we're sort of, until the pandemic ends, constantly going to be in the need for new development, for new antibodies. You take whatever you can forward to treat, but I don't think again, it's not an either or. We're going to try to do both.
This is a question for Marina. I hope, yeah, she's still there. Peter, you and Boris. This kind of perhaps for the CD4 binding site antibodies, kind of rebound without clear change in susceptibility or resistance, and also, Marina, the work that you showed with you know, the susceptibility at baseline really not correlating that well with outcome. I know that Peter and BAN are phase 2, your phase 2 study, not Banner, but your phase 2B study. You're requiring both susceptibility based on an IC something, IC 80 or 90, and an MPI. And it was and, not or, right? So Marina, did you look at the MPI, and did that help in any way distinguish who's susceptible to 3BNC117, and Boris, the same with the kind of rebounding in VRC07. Did you look at MPI, and was that a component of the rebounding virus that didn't appear to be?
Yeah, so I can think, so yes, in some cases, yes. I can think of 1 or 2 participants in the first ATI study with a 3BNC only that we could only explain the rebounding viruses by not reaching a high, you know, a very high MPI. But I also think that it may be that if we have, as we acquire more and more sequences and more phenotypic information, more clinical outcome information, we will get better at predicting CD4 binding site resistance. And I think it's going to be more of a spectrum. So we're going to really need to understand what those cutoffs are going to need to be.
So I think that also comes back to Lucio's point. All these studies are relatively small. That's always our problem. They're not easy to do. You know, people have to participate, wanted to participate, and so we get like 12. Maybe we have like 17 people in one study. Then we have with 2 antibodies. The other study has 12 people with 3 different antibodies. And so I think, I mean, it's great that 3BNC117, 10, and 74 has so many different trials going on, because just accumulative, it's going to generate a lot of information. But I think that's kind of what I think you're pointing to as well. You know, if we just test them in like 10 people and then we throw the next version of antibodies on people, then the question is, do we really learn enough? So I think it's just not enough data. We don't have enough people in these trials to really figure that out, and that it's not predictive, the baseline. I mean, we see the same thing that Marina reported. You know, it doesn't always match. Fully susceptible, great. Not susceptible, not great. It's on and off. And I think that it's just because the sample size is too small. So to define clear pattern, I think we just need more.
It is an interesting thing, though, to see with a lot of antibody on board rebound. And we've had lots of sidebar conversations around it. And one of the speculation is cell-to-cell transmission, being you've got an entry inhibition. If you've got cell-to-cell transmission with low concentration of the antibody intracellularly, is that a mechanism of escape? I'm not a modeler, so I can't really answer whether or not the rebounds really fit that. I mean, there's a lot of speculation on that, which again, may be one of the benefits of looking at a combination of things that act intracellularly versus extracellular. But it's curious to me that this, in particular with the CD4 binding bNAbs, that this seems to be more of an issue, I think, than what you see with some of the other sites on the envelope. I don't know if either one.
Well, that's, I mean, Christina Oxenbauer at UAB has an essay where they actually compare that now. And so what Joe referred to also, we saw these viral rebounds in people after VRC07 523LS treatment at like 100 micrograms of plasma levels of the antibody, fully susceptible virus broke through. A little bit unclear. It was actually not so different than from other CD4 binding site antibodies. So it seems to be maybe a class effect to some degree, but so I was really waiting to see what Christina is going to show, if that is kind of a cell-to-cell spread that is not inhibited well by the CD4 binding site antibodies. Who knows? But I think we have a question here in the back.
That's nowhere near getting to what you just asked. But greetings, everyone. Dazon Dixon Jallo with Sister Love based in Atlanta and in Johannesburg. And I'm also a part of the Women's HIV Research Collaborative at HANC, and as well as the Women at Risk Subcommittee for HPTN. So mine sort of comes back to the ground level in terms of what Sharon Ann is alluding to, what Richard has talked about, and Simon and Joe as well. And that is one of the initiatives that we're working on, for example, is we are the community engagement coordinator team. My organization is the team for the Erase HIV Study for the Cure in the Martin Delaney Collaboratory. And one of the things that we're setting out to demonstrate is the really powerful and important role that community engagement plays at the bench level. And so I think that that's what's also been missing a lot of times in these spaces, is that when we're just talking in the context of the molecules and in terms of the particular, the community of science becomes cells before it becomes people's bodies. And my argument, of course, is that when you engage community before you even identify the cells, that you have a better chance to sort through some of these really complicated, complex, and engaging, exciting questions that, yes, the community is sitting in expectation of you finding some answers to, but there's a large part that wants to be a part of that discovery, that wants to be a part of sorting through some of those really difficult questions. The lived experience is a part of that data. And so I guess my question comes to, what is your community engagement strategy for the work that you have done, even in these phase 1 and early phase 2 studies, in wherever they're happening locally, or even where you're affecting whichever clade you're looking at? How is the community currently involved in the work that you are doing, from the Banner study to the other studies that have been discussed?
I mean, we work with community advisory boards. ViiV is very committed to that. I'd still like to see us bring the community closer early in development. And I think the fact that we're talking about neonates and postnatal periods and all that, and for approval, you've got to prove all the safety data out there. It's just striking the right balance. But there really are, especially in bNAbs, some unique opportunities to move community closer to where we are. So we're engaging more along those lines, and any further input you can provide, and you know Kim Smith and company very well.
Absolutely. Well, the first part, if not obvious, would encourage engagement with community organizations as your partners in the studies, in understanding and building their not only their literacy levels, but their ability to engage, their capacity to articulate, to include and incorporate other ideas from community when you're grappling with some of these questions. I've seen it in the cure space, how there have been parts of studies that have actually been pivoted because of the conversations that are happening at the CAB level. And so I encourage that in this process as well. And I mean, I know people have been looking at bNAbs for a long time. What I don't know is any community organization that's doing treatment, prevention, all the education that's actually in community talking about this as well.
It's interesting that you bring this up. IAVI's shift towards the pediatric space really began through our colleague Shelley Malhotra's engagement of communities in South Africa. And they're the ones that came and said, there is a medical need, there is an opportunity, and the community is actually quite excited about this and allowed us to have that space to say, maybe this is where these antibodies really should be focused. And I think that this past year has been just an evolution in thinking about this, which I think speaks very well to your points.
Thank you.
Speaking as a community member involved in the Rio trial on the data safety monitoring panel, and Simon is on the steering group, the UK Community Advisory Board, which I've been a member of for decades, is involved in all trials that happen in the UK that the British HIV Association is related to. bNAbs, for me, have been something of particular interest, attracted by the safety profile, attracted by the fact that, as I know, ARVs are not titrated according to body mass. Most clinical trials, well, less than 25% involve women for AIVs. We do process them differently. And we do, because we've got a bit of a smaller body mass, tend to have exacerbated side effects. Speaking personally, definitely insomnia. Well, at least very unusual dreams, shall we say, that wake you up. bNAbs, the profile that relates to mothers, babies, children, the potential of a vaccine that would provide safety in cultures and countries where that safety can't be provided, I think makes bNAbs tremendously exciting and tremendously interesting. And as I do communications, it is like what I do, what I've always done. Believe you me, I will do my level best to share information about bNAbs in the UK.
I think that's actually a perfect final words. They are very exciting. I agree very much with the bNAbs. Sarah Fittler actually had mentioned that there are 72 people in the Rio study. That's right, I forgot that. So that's actually, we're starting to get to the larger size studies. And I don't know if Sarah has another question.
Thank you. Sorry to be that person at the end of the conversation. I just wanted to thank Joe for bringing that up, because Joe and Simon, who are in your audience, have worked right from the very beginning of us designing this study together. They have done amazing jobs at working on our website, recruiting participants who actually contact me because they've been talking to Joe and Simon, and through their community groups have said they're interested in the study. So I would absolutely advocate that, you know, paid participants who are part of the community should be part of every aspect of any trial design. And I've really learned that over the years. So we have members on our leadership group, our trial steering committee, and our data and safety monitoring committee, and it has only been a positive experience. So thank you.
Thank you. So I want to thank the panel and the speakers. There was an interesting discussion, and I think now it's lunch. Great.
I'd also advocate maybe next time have Dathan and Joe on the panel instead of me.
Great, Richard.
For those of you online, we will be back at 1:15 p.m. Eastern.
Good afternoon. Hopefully had a nice lunch. My name is Tae Chan from NIID NIH. I'd like to thank Veronica and everybody else for organizing this meeting. So we'll have two presentations first, and then we'll have almost 1 hour open discussion. The first speaker is Nicole Dorius-Rose from the Vaccine Research Center at NIID.
Afternoon. Thank you for coming back from lunch. I'm going to talk about measuring in vitro susceptibility to bNAbs, and this is a topic that we've already been discussing quite a bit, and there's really two places where we need to think about measuring in vitro susceptibility, and one is down selecting products that we want to use for therapy, and the other is monitoring the susceptibility in patients before and during treatment. So hang on. Get rid of this floating controls here. Oops. Okay. So the most common measurement that we use for this is the neutralization assay. And this is really a functional definition. It means prevention of target cell infection by free virus. And neutralization is typically measured with pseudoviruses on cell lines. Now, as Marina pointed out earlier, this doesn't always give you the same answers as you get if you use PBMC-grown virus either on cell lines or on PBMC. And that's actually been known for many years that if you do a PBMC assay, you might get a slightly different answer. But that said, what we typically do is measuring with pseudoviruses on cell lines, and I'll talk about why we do that in a minute. But there are, of course, other functions that can be measured. Blocking cell-to-cell infection was brought up earlier, non-neutralizing functions like ADCC, looking at IgG glycoforms and viral outgrowth are all assays that have been used experimentally to study antibodies. And how do I go forward? But only neutralization has been shown to correlate with in vivo protection. And so that's what I'll be talking about for the rest of the talk today. So I'm going to talk a little bit about just briefly how the assays are done, and then what do the numbers mean? We all look at these numbers, but how do we actually calculate them? What do they mean? So just really briefly, we generally use envelope pseudoviruses. These viruses give us a single round of infection. This makes the assay readout more linear and makes the assay more reproducible. It's not as reproducible as binding assays, but to follow on what was said on one of the earlier talks, we're confident in a threefold change in neutralization. We're confident in measuring that. And one of the benefits of this, another benefit of the pseudovirus is that we can very quickly produce stocks by co-transfection of a standard HIV genome backbone plasmid with the envelope of choice. And so you can quickly get envelopes from patients, or you can have panels of hundreds of envelope strains or envelope mutants, and it's very useful. You don't have to grow large stocks of replicating HIV, so it's a lot logistically easier, and you don't need a biosafety level 3 lab to do it. So very briefly, the way neutralization assays work, and you heard about this before from Jackie, but briefly, you take your virus, you put it on target cells. The target cells generally bear CD4, CCR5, and CXCR4. And there's a reporter gene, typically firefly luciferase, that's either integrated into the cells or it's brought in by the virus, depending on the particular assay you use. And so you apply your virus to these cells, and after 2 days, you get luciferase activity in light, and you measure the light. In a neutralization assay, you would pre-incubate your neutralizing antibodies with your virus, and you apply them to the target cells. And then you don't get infection, you don't get light, and you measure the decrease in light emission. And so you do this with a dilution series of your viruses, and then you quantify this with the IC50 or IC80. So an IC50 is the concentration of antibodies at which 50% of your virus is neutralized. IC80 is 80%. You can also do 90%. And importantly, smaller values are more potent. This is different from what you do when you're looking at serum or plasma, where you do an ID50, D standing for dilution, so inhibitory dilution 50 or inhibitory dilution 80. So it's the reciprocal dilution of the serum or the plasma at which 50% of the virus is neutralized. And in this case, larger values are more potent. And this is, people are often, my students are often confused by this. So smaller if you're looking at IC50, larger if you're looking at ID50. What I didn't include on this slide is MPI, or maximum percent inhibition. For some of our broadly neutralizing antibodies, particularly the ones that target glycans, like 1074, you get a curve that does not go all the way to 100%. You get a plateau value. And in that, we don't actually know what the impact of that is going to be in vivo. But that is something that we measure and sometimes look at. And later on, Yunda Huang is going to talk about a more sophisticated measure called PT80 that we hope has more relevance to the clinical situation. These pseudovirus neutralization assays can be done at various scales. So from a research perspective, the assays can be done manually or using automated systems. They can be small scale in many research labs, making it very handy for basic research. Large scale is what we need to think about for treatment. And that's done in just a few research labs. Our groups at the Vaccine Research Center at the NIH, IAVI, and Duke University, David Montefiore's lab, and Mike Siemens' lab at Harvard. And in particular, the lab at Duke has a validated assay that they've been using with the HVTN. And commercially, this is done by Monogram, as you heard. So Jackie, stop me if I say anything wrong. But their assay is validated to CLIA cap specifications. And they have the PhenoSense HIV neutralization assay can be used for down selecting your clinical product. And they also have a program for pre-screening envelopes for potential participants in the antibody therapy trials, as you've heard about. So I've told you how these assays work, but are we measuring the right thing? Does in vitro neutralization in these pseudovirus assays actually correlate with in vivo protection? In animal models, the answer is a resounding yes. So we know that to get protection from a SHIV challenge in the monkey model and the macaque model, if you have a more potent antibody, you can use a lower dose to get protection. And so I'm going to show an example. This is actually a study we did with Boris, where we had two different antibodies, PGDM 1400 and the CAP256 antibody, that target the same epitope. But for the particular challenge virus we're using, the CAP256 antibody was 10 times more potent, or had a 10 times smaller IC50. And so when Boris gave these animals different amounts of the antibody at the lowest dose, so giving the same dose to all the animals, at the lowest dose, PGDM 1400 did not protect all the animals. 3 out of 4 animals were infected, but CAP256, the more potent antibody, was able to protect 4 out of 4 animals. So in vitro potency translating to in vivo potency. And this has been done in dozens of studies in the NHP model, and a recent meta-analysis done by Yunda and others showed a nice correlation of the ID50, so the serum titers, with protection from challenge. So this is complete protection from infection. So looks great in the monkey model, but what about the human situation? So there has been one real proof of concept clinical trial. And so we've been talking about numbers. This is actually a trial with 4,600 people. And so we've heard about the AMP clinical trial. So this is the antibody-mediated protection trials, asking the question whether a broadly neutralizing antibody, in this case VRC01, could prevent HIV infection. And so this was 2 separate studies actually done together. One of them, and we've talked about target populations. So one of them was done in women in sub-Saharan Africa, and the other one was done with men and transgender women in the Americas, with a total of 4,600 people, as I said. Participants were either given 10 or 30 mg per kg of VRC01 IV every 8 weeks versus placebo. Now, in the study, there was no overall preventive effect, and that was disappointing, but there was a very interesting finding. So envelope genes from all of the infected participants were recovered and made into pseudoviruses and tested for sensitivity to VRC01 in the pseudovirus neutralization assay. And what they saw was that VRC01 actually prevented infection by the very sensitive viruses. So they pre-specified classes of sensitivity to VRC01, less than 1 microgram, 1 to 3, or 3 micrograms per mil. And in that group of viruses that were more sensitive, there was 75% efficacy. So this was considered proof of concept that broadly neutralizing antibodies can prevent infection, and support for the clinical relevance of the pseudovirus assay. So given now that we have an assay and that can be done at a reasonable scale, and we believe it has some clinical relevance, how can we use this data? So as was mentioned before, this is critical for assessing broadly neutralizing antibodies for possible clinical use, for down selecting between different potential products. And so we generally measure breadth and potency on large pseudovirus panels. And so you'll see figures like this where you've got a stack of dots. Each stack of dots is a different antibody, and every dot is a different virus on a panel. So in this example, these were tested on a 208 virus multi-clade panel. And one of these antibodies was also tested on a 100 clade C virus panel. And potency is lower. Potency is better at the lower part of the graph. So we can test numbers of viruses in the hundreds. And then you have to decide what panel do you want to use. So we used our multi-clade panel to see what things would look like worldwide. We used a clade C panel because we were interested in using this particular antibody in South Africa. So this is one of the things you want to do. Do you want to do local or global, single clade or multi-clade? And do you want your viruses to be a standard panel that people have been using for years and years, so you can look at historic data, or do you want more contemporary viruses? And this has come up recently. This is a study by Nono Nkize and Penny Moore's group, where they were looking at clade C envelopes that were collected at different points in time. So the lighter color is ones from the late 90s and then the early 2000s. And then the darker color bars are the viruses from the AMP trial, actually. So more recent contemporary viruses. And what they saw, and then they tested them against a bunch of different things that are being used in the clinic, and found that for certain monoclonal antibodies against certain epitopes, there does seem to be an increase in resistance to these antibodies or decrease in sensitivity. Now you can see there's a big range of sensitivity among the different viruses, but the whole population seemed to be shifting to slightly more resistant, suggesting that we probably want to be using panels of contemporary viruses. We've also heard people mention the issue of clades, and so I want to show an example of that. So this is data from our multi-clade panel, and so we've color-coded the different clades, and what I'm showing is the percentage of virus neutralized at a particular cutoff in each of these clades for different antibodies. So in this case, it's the Captive56 antibody, which is great against clades that you find in Africa and absolutely terrible against clade B. Does not have any activity at this cutoff at clade B at all. Which was a big disappointment to me personally. Another example is 101074, which you just heard about. 101074 has 0 activity against clade AE. So because of that, you would never use Captive56 in Europe or the Americas, and you would never use 101074 in China or Thailand, where AE is prevalent. There are, however, other antibodies that don't have these clade gaps, and so might be more useful everywhere or useful as a partner. So this is definitely the kind of thing you want to do ahead of time, as was mentioned, to make sure that your antibody is appropriate for the population that you want to use it in. We've also heard people talking about using combinations. You don't want to use monotherapy. You want to use a combination of antibodies, but how do you determine how good your combination is going to be? So as some examples have been using antibodies against different targets. So CD4 binding site antibodies like VRC07 or V1, V2, Apex antibodies like Captive56. And so in the data I'm going to show, I don't know how the color is coming out on this, but the way the data is being presented here is each of these lines represents a different virus in a 200 virus panel. Blue represents viruses that are resistant at our particular cutoff, and then orange and red are the sensitive antibodies. And what you can see is that for any given virus, you might have one or the other, or both of the viruses of the antibodies being active. So in this top line, it's resistant to Captive56 but sensitive to VRC07, and then these two viruses are the opposite. They're only sensitive to this one. And so if you think about coverage in a combination in this way, if you want just one or the other or both of your antibodies to be active, then you can get really good coverage. So in this example, the two individual antibodies were 62 or 75% coverage, but if you mix them, it's 90%. And the simple way we do the calculation is for each virus, you count the IC80 of the more potent antibody. And that's how we got this beautiful collection of red and orange. But monotherapy is not what you want to do for treatment. You want to have multiple of your components being active at the same time. So although thinking about it in this way, having one or the other or both is probably acceptable for prevention, it's not going to work for treatment. And that's what we've, of course, learned from antiretrovirals. So what happens if we say that both antibodies need to be active against the virus? Well, then in this particular example, we go down to 47%. So in this combination, although potentially for treatment, it might cover 9%. For prevention, you might get 90% coverage. If you want both of them to be active, you only get 47%. So when you're looking at these data, you need to be careful how you're interpreting the data, the way combinations are being calculated and how you interpret that. Something else you have to think about when you're looking at coverage is what is your cutoff? You have to pick a arbitrary cutoff for what do you call sensitive or resistance. And so the answer that you get is really going to depend on the cutoff that you put in. So in this example, I've got VRC01 and 2 of its engineered derivatives. And we initially, when we do the assays in the lab, we generally set our cutoff at an IC50 of less than 50 micrograms per mil. Why? This does not have anything to do with the in vivo situation. This is how much is convenient for us to put into the assay. Okay. So when we set the cutoff this way, VRC01 neutralizes 90% of the viruses in our 200 virus panel. This looks great. And its engineered derivatives look a little bit better. But what if we set a more stringent cutoff? So based on what we saw in the AMP study, it's been suggested that the cutoff ought to be an IC80 of less than 1 microgram per mil. So if we recalculate our data this way, now we've got less than half of the viruses being sensitive to VRC01. Well, no wonder we didn't get a signal in the VRC01 for efficacy, right? But the engineered antibodies have a much higher coverage at this cutoff and are probably going to work better. And so once we've decided on which ones we're going to use, how are we going to use neutralization data for the treatment trials? And so we heard a little bit about this, that in the broadly neutralizing antibody therapy trials, either in viremics or in the context of analytical treatment interruption, in some studies, the subjects were pre-screened for envelopes that were sensitive to the study bNAb at a cutoff that you set. And this is going to give you a better chance that treatment is going to help. But you might want to not pre-screen if you're trying to do something more exploratory and perhaps to try to figure out where that cutoff ought to be. And then how do you actually do the pre-screening? There are a couple of ways that you can actually assess the envelopes. You can clone and test individual envelopes, and that's probably the most stringent way to do that, because in every person there's going to be quasi-species, and some of those envelopes are going to be more or less sensitive. But how deep do you need to get? Do you do 10? Do you do 100? And it gets impractical when you try to do it at scale. So another way to do that is to do shotgun sequencing and test a whole mix of pseudoviruses, which I believe is what Monogram does. So again, how sensitive you're going to make your test is going to depend on sort of logistics. And so you can do this ahead of time, and also the sequencing and sensitivity testing of rebound viruses can help explain loss of bNAb-mediated control in some of these studies. So I'm just going to really quickly do a sort of an older example. This is from the VRC601 trial, where viremic subjects were given a single infusion of VRC01. This was one of the very first of this kind of trial. And some of the subjects had a temporary decrease in viral load. So in this figure, viral load is normalized to the initial value, and then you can see that in several of the subjects, you got a decrease of 1 to 2 logs in the viral load, and for 2 of the patients, it stayed down for a while. For most of the patients, it went back up, and for 2 of the participants, there was no change at all. And so one of the things the authors did was they looked at sensitivity to VRC01 before and after, and saw that in some cases, at the post-treatment time point, the resistance had increased, or looking at the other way, the sensitivity had decreased. And the magnitude of the change varied from person to person, and it wasn't in everybody, but in several of the subjects, it was very clear that the antibody was putting this selective pressure on the viruses, which could be contributing to the loss of control and contributing to rebound. Okay, so in conclusion, I've shown you that the pseudovirus neutralization assays do correlate with in vivo protection. These assays are essential for selecting clinical candidates, but when you do so, you need to be careful in your choice of virus panels and think about what cutoffs you're using to call things sensitive and resistant. And the assays can be used clinically in therapy trials for pre-screening patients or monitoring the viral phenotype changes during therapy. And so I'd just like to thank everyone who's worked on these things with me over the years and take any questions. Thank you. Are we doing questions now or saving it for this session?
We have plenty of time, so can
Okay.
So there's a question in the chat box.
Yeah.
Read this question.
Okay. So Margaret Gartland asks, I have a question, or actually, Margaret, do you want to unmute and ask your question, or I can just read it? I'll read it. So I have a question about whether we should consider any standardization across different phenotypic assays and across bNAbs, such that we consider including the same reference viruses or panels of viruses in our assays and reporting FC from reference virus rather than absolute values. Well, that's interesting. There are a few, so about the reference viruses. So there are a few panels that people have been using standard, but not everybody uses the same ones. So NIH and IAVI have been using a set of about 200. Mike Seaman has a multi-clade panel and a clade C panel. There's more and more commonly people are using the viruses from the AMP study that were cloned from the placebo recipients, so the ones that are not being selected for VRC01 resistance. So I think that having the same virus panel to compare your different products is going to be absolutely, yes, is very important to have a fair comparison. And then reporting FC from reference virus rather than absolute values, that's something I hadn't thought of doing before. It's something we can consider. You do see that there's a very wide range across multiple orders of magnitude in the sensitivity of given viruses, even within a clade or within a geographic area. So it might be difficult to do that. Second question, how do we define emergence of resistance from pretreatment to rebound? Do we consider a 3-fold change or a 10-fold change of sensitivity to be clinically meaningful? That's something we can only determine by doing the experiments. We've actually been surprised by the level of change that we see in the study that I showed you and in some Tayug studies. There might be a 5-fold change or less, and it's, well, that doesn't look like it's giving resistance, but we don't actually know how that relates. We don't know yet how that relates to the in vivo situation. So I think that doing more of these studies is going to be needed to give us a big enough body of data to actually answer that question. The other issue is not just change. We can look at change in sensitivity, and we can also look at absolute sensitivity if it turns out that there is a clinically meaningful cutoff, which I hope there will be, because that will make our lives easier in trying to do these kinds of studies.
Any other questions? Okay. Thank you.
All right, thank you.
The second presenter is Yunda Wang from Fred Hutchinson Cancer Research Center.
Good afternoon, everyone. So first of all, thank you, Veronica, and then the forum for inviting me to speak about this topic that I'm really excited about. So you need to switch your brain a little bit. We've been talking about treatment for a while. I'm glad that Nicole already primed us a little bit. I'm gonna talk. The first half of my talk will be focusing on prevention. But my hope is that whatever we learned in the AMP trial for HIV prevention can possibly be a learning experience for what we can do for treatment. So thank Nicole for giving a little bit background on AMP. So after that, I will primarily talk about using neutralization biomarkers for predicting efficacy, prevention efficacy, or control viral load. So the first bullet for bNAb-mediated prevention will be based on the AMP trial, the antibody-mediated prevention trial. The second bullet is also based on the M trial, but in that study, we have had participants acquired HIV infection during the study, and we looked at the viral load after HIV acquisition and see the effect of VRC01 on it. So I will show a bit data on that. So this is AMP for prevention trial conducted in 4,600 individuals with 10 infusions of VRC01 with a low and high dose of VRC01, 10 mg per kilo and 30 mg per kilo with a placebo control. And this is also the same results in a slightly different format that Nicole showed, where there was no overall prevention efficacy. On the right panel, as you can see, the upper curve, where against VRC01 neutralization sensitive viruses with IC80 defined as less than 1 microgram per ml, you do see a roughly 75% efficacy of VRC01 preventing HIV infection. And whether or not, so we also saw in that data that we saw the prevention efficacy actually smoothly decreases with IC80, with the susceptibility we've been talking about in the morning of VRC01 against acquired virus in M participant. Basically, for all M participants who acquired HIV infection during the study, we isolated the virus and made them into pseudotype and measure their susceptibility using IC80 that Nicole spoke about. And we were able to analyze the relationship between the prevention efficacy with the IC80 value on the x-axis. And you see this decreasing trend where PE was estimated to be 4.5 times higher against viruses that are tenfold more sensitive to VRC01 neutralization. More specifically, for example, we estimated 81% efficacy against virus with IC80 of 0.2 microgram per ml, but the efficacy was much lower, 16.5% against viruses with a susceptibility of 2.0 microgram per ml. However, my point is IC80 is not sufficient to quantify the ability of a VRC01 recipient sera to neutralize an exposing virus at a given time. Why? The reason is IC80 is a measure of in vitro activity of the bNAb against a virus in the lab. It has nothing to do with the concentration of the bNAb in a VRC01 recipient. So for that reason, we propose using the so-called PT80. It stands for predicted ID80 titer, the ID80 concept that Nicole just introduced. So more specifically, it's a similar shape, but you kind of revert it. So on the y-axis, you'll see the prevention efficacy from the AMP trial. On x-axis, now you see the predicted ID80 titer. I will get to what that means later on. But over here, you see, you can ignore the two dotted curves, but you see the three colored curves, one blue, one red, four curves. One blue, one red, one green, and one black. The one in black is the one that Nicole also showed for the AMP trial participant, where you see this increasing, the PE increases with higher predicted ID80 titer. For the other curves showing the curves from the study, actually Nicole also mentioned, where 256 animals had gone through the challenge study with infusions of 16 different bNAbs targeting different epitopes on the trimer and had different challenge viruses. The message here is that overall, you see a pretty consistent relationship between the prevention efficacy and PT80 on the x-axis, no matter what bNAbs targeting, what epitopes you're looking at, no matter what challenge virus you're looking at, and even in a human trial depicted by the black curve here, that exposed to any kind of viruses. As mentioned, the trial was done in two different regions, the Clade B region in North America and Europe, and then the Clade C region in Sub-Saharan Africa. So this is aggregated across all viruses of different clades and regions. So PT80 is not actually fancier. It's actually easier than ID80 titer. ID80 is something that you need to measure all the time in the lab. You have to get a serum sample from the participant who received the bNAb, and you measure it against the virus neutralization activity. PT80 is something that, well, maybe you don't have to do that. So it's a proxy of the experimental measure ID80. How do we do that? I'll get to that a little bit later. But here I want to show you that PT80 is a good proxy of the experimentally measured ID80 titer. As you can see on this graph, this is shown for VRCO1 based on the AM trial data. On the y-axis shows the observed ID80 titer. We basically get a serum sample from the VRCO1 recipient, measure it against the ID80 titer against the viruses that you see on the columns. And on x-axis here is predicted ID80 titer. And you can see all the dots are pretty much on the diagonal line for all the viruses shown in the column. And there are rows, that means different approaches where you use to predict the ID80 titer. I circled the first two approaches that only use information from the concentration of the bNAb in the serum sample of the participant and the IC80, the in vitro IC80. So those are the two approaches that are the easiest that you can also very reasonably predict the experimentally measured ID80 titer. The other two approaches, which I won't get into, uses more complicated PK/PD modeling that also needs to have the ID80 titer measured at a certain time points. If we just focus on the first two approaches, you just need the concentrations and PK model and IC80 to be able to predict PT80. So similarly, we also saw similar data patterns for the other bNAbs. This is a slide showing for VRCO7 5-FLS, which we talked a lot about in the morning. Here also shows, you see the dots lining on the diagonal line. PT80 also predicts well the experimental measure ID80 titer. And this is for another bNAb, PGT121-414LS. You see the same pattern. And this is for PGDM1400LS. On the y-axis, on the y columns are the viruses. Oh, actually, each panel is one virus. You see all the dots on the diagonal line shows the same message. And this is another slide showing, similarly, we assess the predictability of the predicted ID80 with the observed ID80 titer. You see the same pattern for this combination, this triple combination. So with CD4 binding site, with V3, V1, V2 targeting bNAbs, you see the lines are pretty much on the same on the diagonal line, and you see different colors basically indicating different ways of predicting the combination ID80 titer, because now we're talking about multiple bNAbs. But no matter which method you use, assume additive effect, Bliss Hill model interaction, they pretty much give you similar pattern that we can do a pretty good job predicting ID80 titer using concentration and IC80. One more, this is a dual combination, same pattern. So, okay, so now get to the specifics. How do you actually calculate this PT80? It's simple if you just want to look at the first two approaches, which does a very good job already. So PT80, the good thing is you can calculate PT80 at any given time point where you don't have to draw blood from the participants to measure it experimentally. So what you need is basically, this equation is very simple. One thing divided by the other. The first thing is you need a concentration. So this concentration can be experimentally measured the concentration of the bNAb, or you can build a pretty good pharmacokinetics model to predict the concentration at any given time. So you have a concentration, and in the denominator would be the IC80 that we've been talking about all morning about susceptibility of a bNAb against a certain virus. Then you get PT80. That's the simple way to get the PT80. So this is an example where if you have a concentration of the bNAb in the serum sample of 50 microgram per ml, and if your IC80 is 2, then you get a 20 PT80 of 25. Lucia was just asking me whether it's possible to get a PT80 of 2000. Yes. Well, if you have a concentration of 200 and you have a bNAb against a sensitive virus of 0.01, 200 divided by 0.1, you get 2000. So that's how you get PT80. And then so you can do the same thing for combination bNAbs based on different interaction models. So just to summarize, IC80, what we talked about is the in vitro neutralization property of the clinical lot of a given bNAb against a given virus that Nicole spoke eloquently about. But it's not a sufficient correlate because it has nothing to do with the bNAb concentration in a recipient serum at any given time. On the other hand, PT80, it shows the in vivo neutralization property of the serum sample of a bNAb recipient at a given time against a given virus. So it could be a sufficient correlate, a property of both the bNAb as well as the concentration in the virus. So now that was for prevention context, and then we established PT80 as a correlate of prevention efficacy. Now, within the same trial M study, as I mentioned, there were about 200 participants that acquired HIV infection out of those 4,600 participants during the study. So among those participants who acquired HIV during the study, we measured the viral load over time. What's shown here, on the y-axis is the viral load that measured at diagnosis visit, the first detection. And it's broken down by categories of IC80. So you can see the first 3 bars are for IC80 if a person acquired a virus with IC80 less than 1. The right side 3 bars are for if a person acquired virus with IC80 greater than 1, resistant to VRC01. So if you focus on 3 bars, you actually see that HIV viral load at the time of first detection was lower. You see the 2 green ones, the blue one, sorry, red, colorblind. So these ones have the viral load lower. These are from the VRC low dose group. This is from VRC01 high dose group. And the blue one, this is from the placebo. So you do see a lower viral load compared to placebo for those for those who received the VRC01 if they were exposed to a virus that's sensitive to VRC01. On the other hand, you don't see this kind of effect, did not seem to influence the initial viral load at detection among participants who acquired more resistant virus. So, so this is a work that being led by a few of my colleagues at Frankfurt Cancer Center within the HIV Vaccine Trials Network, where we try to model the HIV viral load of those participants who acquired HIV during the M study. On the upper row here shows the VRC01 concentration on the y-axis over time for these participants. In the second row shows the viral load on the y-axis over time since the time of the first positive. You see we did a little mathematical modeling of the viral load trajectory. And if you link the two, we actually were able to find that. So here, let me walk you through this. So basically on the y-axis shows the percent of neutralization or percent inhibition for neutralization. So this curve shows the in vitro relationship between the concentration of the drug, the PT80, and then the percent inhibition. This curve is what you see in vivo in the M trial. Basically what it's saying here, you have to shift to the right. Okay, you need to shift to the right. If you wanted to neutralize 80% of virus, in vitro, you need, for example, 1 microgram per ml. In vivo, you need about 600. So that scaling factor is about 600 for controlling the viral replication. So in parallel, that number for prevention is about 200. Actually, 200 is for 90%. For 80% is 125. So it's about 100-fold scaling for prevention. But for this, for potency in viral reduction, the scaling is about 600. Of course, this is based on very limited data. And then for all these M participants, we had the viral load measure only before the ART initiation. So they normally have about only 3 time points after diagnosis. So we don't see viral rebound and so on. So this is primarily based on, this limited data you see on this curve. Okay, so in summary, we found that PT80 predicted ID80 titer predicts well experimentally measured ID80 for both single and combination bNAbs. So we think it's an important tool to use it to design future bNAb trials and to evaluate PT80 as a correlated prevention efficacy. PT80 can be used to rank bNAb regimens by the potential prevention efficacy because bNAb regimens with greater PT80 levels are predicted to have higher prevention efficacy. And we, based on the limited data we have from MAM, compared to prevention, with it looks like higher PT80 will be needed for viral suppression via HIV bNAbs. So here I wanted to thank all the study teams and study participants, especially, and my colleagues from the HVTN Data Center, as well as the lab colleagues and Leadership Operations Center. And I also like to thank our founder, NIAID, and our wonderful, wonderful collaboration through all these years with our VRC colleagues over here. Thank you.
That was a great presentation, and obviously a huge amount of work that went into that. I guess I have a comment and a question. I mean, in some respects, the PT80 concept seems analogous to what we used to call the inhibitory quotient, where we were looking at how much drug was achievable at trough in the plasma, and how did that relate to the susceptibility of the virus. I guess the question I have is, I can see how the PT80 is useful in selecting antibodies for study, but it's not clear how it can be used to select individuals as participants.
For treatment, right? Yeah, for treatment, I would still think, the discussions that we had in the morning, looking at susceptibility, is the most valuable one, yes. But if you wanted to look at when to redose the participants, you do need to know the concentration in addition to the IC80. Yeah.
How does the PT80 deal with viruses that escape because of the MPI, where the IC80 might not shift very much at all?
Yeah, that's a good question. PT80 doesn't deal with that, but actually I didn't present this. There are other measurements, for example, IIP, where it does incorporate the slope IC50, IC80 value, that we try to look at IIP to see whether it also predicts prevention efficacy. It actually doesn't do as well a job as PT80 in this particular data set.
Sure. And in the AMP data set, you actually use the participants virus. In those prevention trials where they were PK trials, you must have used some external virus, right? Because those weren't prevention studies. The HPTN, PTN, combination antibody things that you use some standard virus for calculating the PT80, right? Is that right?
Yeah, that's what Nicole touched on as well. So right now, the figure that Nicole showed were led the study where you see a little drift over time. So right now, the one on the right panel, or on the right bar that she showed is the viruses acquired by placebo recipients in the AMP trial. So we use that kind of as a standard, as a more contemporary reference panel. Yeah.
Great, thank you.
So I have one, following up on Dr. Kurtz's question. So for the treatment studies, what's the practicality of the PT80, knowing that everybody's a different IC80 values, and people can predict the PK of most of the antibodies that are being used in the clinical trials. Yeah. They can predict how long it's going to last and at what point was the concentration. It's all up to susceptibility of the individual virus to given antibodies, right?
Yeah. So again, so the question like Dan raised, and then like the figure that Peter showed in the morning, right? You have 3 different curves for different IC80 values. So I would imagine if you use your x-axis, basically calculate the PT80 using the value you already have, you may just get one curve. You see a consistent relationship between your viral control and PT80. And then so you can see, when the PT80 gets to a certain low level, then maybe it's time to redose, or you have a higher concentration. You need a higher concentration to boost up your PT80, because as you said, the IC80 against the same virus doesn't change for the same individual. Yeah.
So I guess we have our speakers out here, and Dr. Julia Lathrop will join the panel. any questions? Okay. So I was given a pre-drafted questions, and this session was really about